Hello. All be testing. Sound check. Chintan.
યસ યસ
અ,
અવાજ આવે છે ક્લિયર.
અને સ્ટ્રીમિંગ ઓન છે.
યસ સ્ટ્રીમિંગ ઇઝ ઓન.
कैमरा में जरा पोडियम दिखाओ. बेटा जरा पोडियम दिखाओ कैमरा पे. हेलो. All be testing. Sound check. वीडियो नहीं आ रहा है. यहाँ पे नहीं है. ओके ओके लाइव आ गया. ठीक है. That's all. वो बोले
Good morning, all. A very warm welcome to the first parallel session in Hall B. Calcutta has long been the home for the most dramatic advances in understanding the cause and nature of cholera. While Robert Koch's isolation of the bacteria from the autopsy sample from a 22-year-old patient from Campbell Medical College and SND's discovery of cholera endotoxin were a milestone, the oral cholera vaccine of Eltor biotype developed by Shoti Mohan Mukherjee was first tried in human volunteers in Calcutta. In terms of cholera surveillance, immunobiology, and vaccine development, this study has left a lasting legacy for us. In this session, we will be discussing the progresses made on cholera surveillance and immunobiology. There are 5 speakers in this session. Their talk time will be for 15 minutes. A reminder will be given at the end of 12th minute. To lead the session, I call upon stage Dr. Amanda Davis, John Hopkins Bloomberg School of Public Health, and Dr. Gagandeep Ram, CMC Vellore, to lead this session. Please, ma'am. Yeah, it's fine, ma'am.
Thank you.
I hand over to you to
Okay.
call the speakers. Yes,
Right. So then we're going to sit down there, right? And it's my pleasure to call our first speaker, who is my co-chairperson. And she's going to be talking to us about whole genome sequencing of Vibrio cholerae, looking at subcontinental transmission patterns.
Do I? Okay. Will it just show up? Oh, there it goes. Are we ready? Thank you, everyone, for coming. I know there are several really interesting parallel sessions. This was the title of my talk when I was originally in the genomics section. So I gave it a more appropriate title, given that we're talking about cholera surveillance, and it's a little bit more my alley anyway. So we're talking more about exploring new sample types to improve the genomic epi of cholera. So genomic epi basically encompasses population to individual contexts and outbreaks. And in order to contribute to the genomic epi of cholera, we need to improve our access to not only genomic data, but the metadata that's associated with it. So we need improved surveillance so we can understand the exposures and the links between cases. And we need improved diagnostics so that we can rapidly identify cases, preserve samples, and ensure that they are sequenced. Unfortunately, cholera, as you all well know, typically occurs in areas with limited laboratory and surveillance capacity. So our approach to this is to consider low cost and simple tools to use for surveillance and lab. And our focus is primarily on filter paper and rapid diagnostics. So the proof of concept for use of filter paper was performed in Cameroon while we were looking at the epidemiology of cholera in Cameroon. We started this in 2013 following pretty severe outbreaks in the Lake Chad region from 2009 to 2011. And we thought that Lake Chad would be a reservoir for cholera in Cameroon. And we applied simplified surveillance methods from Bangladesh, where we conducted intensive surveillance every 15 days, both for environmental and clinical samples. We brought back sphere swab methods and put gauze in basic water bottles like we have at the back of the room. And we would filter a few liters of water through the gauze and enrich the gauze and clinical stool samples in alkaline peptone water, then RDT them and spot them on filter paper. We showed that the enriched RDT has nearly 100% specificity and that we could then use the filter paper to PCR confirm our findings. And we did detect an outbreak early on and closed off a water source. So in general, the proof of concept was quite successful. And since then, many of my colleagues have begun using filter paper for this specimen preservation method. So then we chose to see what more we could do with the filter paper. And initially we looked to MLVA. I put the abbreviation definition there in case I couldn't remember it. Multiple locus variable number tandem repeat analysis, very long. But basically you're using a PCR platform to focus on 5 gene targets, and then you sequence those looking for varying nucleotides. And those repeats are used to differentiate genotypes at a more local scale. And we looked at 75 specimens from filter paper. We brought in some samples from collaborators at the time in Mozambique and Philippines. And it wasn't groundbreaking science, but we showed that we had sufficient DNA to conduct this analysis. And we found some interesting overlaps in genotypes between Mozambique and Cameroon. We've since used MLVA in several other countries from Nepal, Tanzania, Nigeria, and Kenya. These papers are forthcoming. They were waiting on sequencing results, which is kind of the crux of the story here. So we have been working for some time to try to sequence or optimize sequencing from these specimens. Initially, we worked with Sanger and Nick Thompson's team, led by his postdoc Angel, and we sequenced some of the isolates from the Cameroon outbreak, where we had paired APW enriched and isolates spotted on filter paper. And the figure here shows, I don't have a pointer, but the figure shows that while there was more Vibrio cholera specific DNA in the isolates, as one could imagine, we had larger contigs and DNA coverage to the reference genome on the APW enriched specimens. And basically what that says is that it was successful on both types, and there was promise to improve our sequencing abilities. But one interesting aspect of this that as I've been working to optimize this method presents to me is that if you look, we attempted to do metagenomic analysis of those samples as well. We used Metaspade and we paired the APW enriched and the isolate specimens side by side. And if you can see, 2 I circled, for example, at the top you see on the right side is the isolate, and predominantly you're only seeing cholera, but on the left side of those circles is the APW enriched specimen. And you see Shigellas and E. coli's present, obviously pathogens that like alkaline peptone water, which has a pH of 9.6. But to me, what it says is what else is present if we look at the whole stool sample, and could that play a role in mild to severe disease? So one outcome of this was that we began spotting whole stool on filter paper, and we're trying to improve how we can look at these specimens. A second paper, we performed sequencing on isolates on filter paper. And again, not rocket science findings, but 2 important outcomes were that the MLVA that was conducted here complemented the whole genome sequencing with very similar findings of distinct outbreaks or introductions at 3 different times in Tanzania. But the big takeaway from this was that this was actually possible because we used the isolates spotted on filter paper. We struggled to get the isolates out at the time. It was pre-COVID. There weren't sequencers everywhere. Oh, thank you. And so John Mwaba came to Johns Hopkins and he did the MLVA and whole genome sequencing during his PhD studies. So it was amenable to everybody that the specimens were transferred in this way. There's no biohazard risk, and some of the concerns about sharing isolates were alleviated. So this, I think, included only 20 isolates sequenced to match the 60-something MLVA. But again, another successful attempt at whole genome sequencing from filter paper. And to bring it to the original title, our current analyses are looking at Southeast Africa, exploring that region and subcontinental transmission, and also to explore different sequencing qualities across the different specimen types. We often only get samples from large outbreaks, and filter paper can be used as a means to kind of fill in the gaps that we have in transmission data. For example, in one of our studies in Tanzania, there was an outbreak in Ruvuma, and the MOH found out about it about 7 days after the outbreak had begun, and our study team deployed with the rapid response team. They were there for about a week, brought back 26 isolates, but only 3 grew. However, they had the filter papers they'd spotted and brought back that they could fall back on and that we were able to work with. So in this data set, we had 122 specimens from Kenya, Tanzania, Uganda, and Zambia from 2007 to 2019, and 20 from Malawi that were sequenced on Nanopore. The first were on Illumina. I apologize for skipping that. And then we combined that data with 1,391 published genomes. And the map on the right just shows the distribution of where in each country we have where each specimen came from. And some are missing from this map because we only had country level data. Again, that metadata problem I mentioned earlier. So in the end, we had 98 specimens successfully sequenced through Illumina and 20 on the Nanopore. But some of the interesting findings we found T1 and T5 transmission samples in 2015 in Kenya. We found several of each, and they were last reported in the late 90s. And more interestingly, we found 9 isolates between 2007 and 2011 belonging to the T13 lineage, which might suggest an earlier introduction that was previously estimated. And this table is just showing the lineage distribution that we found in these sequences. As you can see, there's a lot of co-circulation in this region of Southeast Africa, which is kind of unique. In previous studies by my collaborator, Shirley Wall, for example, T12 tends to dominate in Central West Africa. But here, as you can see, for example, Malawi, T11 and T13 are co-circulating for some time. And T11 was previously, I think, only identified in Mozambique, though it's not surprising, given their border, that it's also in Malawi. And T10 and T13 is co-circulating in Tanzania, Kenya, and Uganda throughout this time frame, and Zambia. And this slide is my favorite, this shows just the samples from this Southeast Africa analysis, not those first 2 papers and not an analysis we just finished on Nigeria and Zanzibar. But in it, it demonstrates that from the crappiest stool sample, direct stool on filter paper, to QIAGEN isolates, which are the gold standard, that we were able to successfully sequence and have pretty high percentage of the reference genome covered. And they, at least 50% of stool spotted filter paper were greater than the median coverage depth. So it shows some potential if we continue to optimize that these can be usable sample types. This is just a very preliminary metagenomic analysis of the Southeast Africa data. It's using Metaflan, not Metaspade, so it doesn't take it past the species level, but it just shows the same type of isolates specific to Vibrio cholera, but on the bookends of it, you can see the whole stool on filter paper shows more diversity in the pathogens present, including several ICP1s, which is of interest to our study as we look at choleraphage. So the conclusions are that our findings are limited by the number of isolates and their geographical and temporal distribution. But we did find some evidence of interesting introductions or lineages present when we previously hadn't thought they would be. So if we are able to find more specimens from these timeframes, or more recently, we can contribute to this analysis and better understand some of these transmission events. We hope to complete the metagenomic analysis and also look at the AMR profiles through sequencing and compare to the actual AST done in the labs where that metadata exists. In Kenya, for example, they keep very So we're hoping to do some of that so we can have a better understanding of what AMR can do through sequencing. And of course, we have established a cholera consortium with the Africa CDC, and we hope to continue to fill in some of the gaps in the transmission puzzle through that. I think my time is up, but really quickly, we are also hoping to use RDTs in this specimen preservation. We demonstrated that you can just extract the DNA from the wick of the RDT, and you can confirm PCR in that way. So it eliminates the need for another specimen type like filter paper. So it could potentially make the RDT kind of a one-stop shop for screening and confirmation, whether be enriched, where we showed great specificity, or PCR confirmation, and then perhaps for sequencing. And we're working in collaboration with Andrew Asman's team in Uvira to improve the quantity and quality of DNA that we're extracting from RDT. And so hopefully we can provide results next meeting on how well that worked. So just to take a moment to acknowledge the very many people that have been involved in there. And I realize I actually missed some people as I look at this, like Nick Thompson, as I showed, and Angel. But thank you to everyone that was involved.
Thank you very much, Amanda. We're going to be holding questions until the end. So I'd like you to make sure that you don't forget the questions. And we'll come back to Amanda at the end of the 5 talks. Our next speaker is Sonia Hegde, and she's going to be talking to us about seroincidence of Vibrio cholerae using longitudinal data from Chittagong.
Okay, I guess my time will start. So I'll be talking to you today about a study that we have in Sitakunta subdistrict of Chattogram, Bangladesh. This is a massive team effort, and so I'm just a single person presenting on behalf of many people. So to start, cholera burden estimation relies on passive clinical surveillance. And so if we look at, which is often, and we often miss infections due to variable clinical presentations and different healthcare seeking behaviors. But if we look at this image here and we start on the left-hand side, we know that in a population, there may be many people infected with cholera, but only a certain number of those are symptomatic. And among those, only a certain number seek care. And among those, based on the diagnostic that we use, only a small percentage are identified and reported as cholera. And so we can think about this as a paradigm or cascade of how we capture true disease burden. And this can largely be categorized into 3 different types of data. One is clinical surveillance data. So, right, who's being identified and reported as cholera. The other is healthcare seeking. And the third is serologic data. So trying to get at every infection in the population. And so what we tried to do was to understand how to estimate the true burden of cholera by combining these 3 components. And so just to reiterate, those 3 components of data are serology, healthcare seeking, and clinical surveillance. And so we enrolled a cohort in Chattogram, the Chattogram district in Bangladesh. And we chose this area based on previous sera incidence data because it was a high risk population for cholera. And so we chose the Sitakunta subdistrict, which is just north of the Chattogram city. And in that population where we enrolled a longitudinal cohort to collect serum samples, we also systematically tested medically attended cases at 2 public healthcare facilities. And we systematically tested using RDT, PCR, and culture. And so we started this in January of 2021, where we collected clinical surveillance data and conducted 3 serosurveys in the same population. And of note, because of COVID-19 lockdown, in May of 2021, we had 2 separate populations, time periods of enrollment. We call them round 1A and round 1B. And for this presentation, I'll be showing results from that first round of enrollment. So those participants we enrolled in that round 1A and followed through all 3 serosurveys. And so with that data, both the clinical surveillance and the serosurveys, we were able to collect data on cholera burden over the course of 1 year. What this figure shows here is the time on the x-axis, and the bar plot shows the number of suspected cases enrolled into our clinical surveillance, disaggregated by RDT positive or negative. And then the bars on top of the plot show the timing of our serosurveys. And so collecting this data, the burden data over the course of 1 year, allowed us to look at potential infection windows to estimate seroincidence in 3 different periods. So we could look at before round 1A in the 6-month time period pre-baseline. That's what we're calling period 1. We can also look at period 2, which is the 6 months prior to round 2, approximately the same time period between the first and second serosurveys. And then period 3, which is the 6 months between the second and third serosurvey. And so how did we measure seroincidence? Well, we thought about it in 3 different ways. 1, looking at a fold change in the vibrocidal titers between surveys, so between those time periods I just mentioned. 2, kinetically correcting the vibrocidal titers to better understand the individual titer trajectories to estimate infection. And 3, looking at the cross-sectional serologic profile to estimate seroincidence. And so I'll go through these methods one at a time. So this first one, looking at the fold change in vibrocidal titers, typically vibrocidals, as most people here I'm sure know, are used in immunogenicity studies and other studies to estimate serological incidence of cholera. And it can be usually looked at as a 2-fold or greater than 2-fold change, 4-fold change in titer values to say that someone had a seroincident infection. And so what this figure here shows you is the time during our study period on the x-axis, and on the y-axis is the log vibrocidal titer values. And so you can see that people's vibrocidal titer values change over the course of the period. And so these arrows point at when we did our serologic surveys, and the colored lines you can basically show how those titer values change across those 3 serosurveys. And so just simply estimating the fold change in those periods, so from the first serosurvey to the second and the second serosurvey to the third, we get an estimate of seroincidence of 6.5% in that period 2 and 4.3 in period 3. But we know that vibrocidal titers decay over time, and therefore looking at a fold change in vibrocidal titer values may actually underestimate seroincidence. And this figure here shows how the vibrocidal titers decay over time if you compare the decay to just a baseline titer value. And so to correct for that, we then looked at this method, the second method, which is kinetically corrected vibrocidal values, which basically means we took that decay rate and we used data from the epidemic curve. So I had mentioned how clinical surveillance data can also be used. And then we also used the vibrocidal titer values from each round from the serosurvey. And so using those 3 things together, we then estimated probabilities of infection. And so what this figure here shows you is, it's a bit complicated, but there's 8 different phenotypes. So because we had 3 different serosurveys, there's a possibility of an individual being infected at each of those time periods or not being infected. And so these 8 phenotypes show you every possible combination of that. So to explain it better, if we just look at this first panel, the idea is that, so on the x-axis, we're looking at time over the study period, and on the y-axis are the log titer, virucidal titer values. And the color indicates the probability that the person is infected. And so in this first panel, the phenotype is showing that some person is never infected during our time period. And what this basically shows is that with these lighter colors, with the lower titer values, that our model is working well because we have a higher probability at those lower titer values that the person was never infected. Similarly, if we look at the second panel where we're assuming that somebody may be infected by that third serosurvey, you can see how the lighter green lines basically increase by the third time period. And that also indicates that we have a high probability that the person is being infected by the third round, which mimics the titer values. And so if we look at our estimates of seroincidence by those same time periods, you can see that they're quite different from the fold change values. Again, because now we accounted for that virucidal decay. So then our third method was that we looked at the cross-sectional serologic profile. So previously in our research group, Dr. Andrew Asman had developed a random forest model. So to basically a way to use cross-sectional serology to estimate cholera seroincidence. And so we used those similar methods on the data from this study. And what were those methods? So we basically took titer values from 8 different lab assays. So the virucidal by serotype, Ogawa Inaba, anti-CTB, IgA and IgG, and anti-LPS, IgG, IgA by serotype. And we took those titer values and we input them into this previously developed random forest model, which identified basically whether a person was recently or infected or not. We then took that and we corrected it for sensitivity, which we know sensitivity of the random forest model, because we know that sensitivity decays over time. So your likelihood of identifying whether a person is infected or not decreases as time from infection goes on. And so we corrected for the sensitivity decay, specificity, and a daily risk, assuming that there's a daily risk of infection dependent on the epidemic curve. So that clinical surveillance data that we also collected. And so those things together allowed us to estimate seroincidence. And so now if we look at those values and compare them, you can see that they're quite similar to the kinetically corrected virucidal estimates of seroincidence. And specifically, if you look at the second time period, for example, so seroincidence from the first round to the second round, the seroincidence of 42.6% compared to 42.8% is quite similar by those last 2 methods. But if we go back to that cascade of how we estimate true burden, I think it's important to remember that I only showed you methods using 2 types of data. So the serology and the clinical surveillance data. And so we're currently working on a model that also incorporates the healthcare seeking data, but so that we can estimate the medically attended case to infection ratio. But without the healthcare seeking data, we can still use the results that I just showed you to estimate how many infections there are to a single medically attended case. And when we do that, you see that from the first round to the second round, you have a medically attended case to infection ratio of about 1 to 523 to 3,448, so quite high. And obviously for the fold change method, it's much lower. And then from the second round to the third round, you have a medically attended case to infection ratio of 1 to 48 to 1 to 333. And so you can see it's quite dependent on time period. And again, this is dependent on our clinical surveillance data and the fact that there weren't as many cases in that second time period. And so what to kind of take away from this? So estimates of sera incidence from that, the kinetically corrected vibrocidal method, plus the cross-sectional model are similar. So this would indicate that cross-sectional serologic data can be a useful method for estimating sera incidence. And also that medically attended case to infection ratio is quite high. And so there are many cases or many infections in the population that we are likely missing in clinical surveillance. But with cross-sectional serology or just collecting serology data in general, you can actually get at that. And furthermore, for this type of study, it's a quite unique study because we were able to collect all these different types of data. So the healthcare seeking, clinical surveillance, and serology. But in truth, this can't feasibly be done everywhere because it's quite cumbersome. But so what does the study do? It provides a deeper understanding of that burden estimation cascade. So that figure I showed you in endemic settings specifically, and helps us to interpret how sera incidence data can be used. And even though that may likely differ across settings, the results from the study can still be useful elsewhere. And so with that, I think I just made it in time before the buzzer. I'd like to thank our collaborators and most importantly, our study participants without whom this would not be possible. And also our collaborators at ICDDRB in Bangladesh and our funders at the Bill and Melinda Gates Foundation. Thank you.
Thank you very much, Sonia. Again, a reminder to hold your questions to the end. And to the speakers, don't be too nervous about the buzzer. That was kind of obvious with both Amanda and Sonia. It's a small group and we want to make sure that there's enough time to hear everybody and talk to them. So our next speaker is Ranjan Nandi. He's going to be talking to us about immune responses to oral cholera vaccine in India.
Very good afternoon. We are in Kolkata now, and from the organizer side, I'm as a part of the committee. I welcome you all also for this, and we will be having some exciting lectures, I hope, during these conferences. And now my talk is immune responses to oral cholera vaccine studies from India, from Amanda and also Sonia. Whatever we have seen is actually it's a study, it's a data, it's a collaborative study, it's a lot of manpower involved. And then the specialization, some are field workers, some are lab workers. So I'll acknowledge all of them at the end of my work, talk. I'm Ranjan and Shuman is also here. Probably most of you know that Dr. Kanungo, who is actually taking care of the field, and Dr. Shanta Datta, the director and organizing chairperson of this conference. The disease you all know, but I thought I put one slide because there may be some person from non-cholera person. So it's a life-threatening disease. Even in Kolkata, if I ask even my family members, do you know the cholera? They will say no. Because we coin them as some anthrax and other diseases, but when you go for testing, it's Vibrio cholerae. It's Vibrio cholerae 01 and 139 who is responsible for cholera until unless we have outbreaks. Until unless media is putting an hype, we'll not be talking about the cholera. And in India, when we are trying to push the cholera vaccines, we need it, government is listening. but no further movement. So this is Vibrio cholerae O1 is a classical LTOR. When we talk about the genomes, when we talk about the serology, we are actually talking about this kind of biotypes, and we are seeing that there are mixing of biotypes happening in different strains, and then the classical convention, the canonical convention, the classical LTOR is also moving away, fading away slowly, and this have the serotypes Ogaba and Inaba. So we look into those serotypes. So this is a classic slides from Jan Holmgren. So cholera, the organism doesn't invade. It's a non-invasive pathogen. And this pathogen, of course, invades mucous layer, but it stays on the epithelial surface, but it causes diarrhea due to the toxin. When we talk about the protection, when we are actually looking at here, the preventing the colonization potentials and preventing the cholera toxin action. So basically, in terms of immunity, when you look for the vaccine or from the clinical cases, we are looking into two different kinds of immunities. These are antitoxin immunity, another is antibacterial immunity. So antitoxin immunity is against cholera toxin, but it's not very effective for long-term protection. In case of antibacterial immunity, it's a very important for long-term protection. It is serotype or serogroup specific, like O1 and 139. If we immunize with O1 and then may not protect, it would not protect the 139 or vice versa. And when we talk about this protection, we are talking about the local immunity. But when we measure the serum, like Sonia was telling, is actually serum immunity, which is nothing to do with actual protection. So we are working with indirect marker, and the best indirect marker, what you consider is the fibrocidal titer, is a lysis of the Vibrio cholerae cells with the antisera in presence of complement. We have a unique opportunity because the SANCOL, the oral cholera killed oral cholera vaccine, actually initiated at Kolkata long back in 2006. We continued with the study, safety, immunogenicity, and long-term protection. And it is shown that it showed the 65% protection after 5 years. And when you come back to the fibrocidal titer, after 5 years, it is went up because it's rapidly when it rises and then goes down. And the vaccine was pre-qualified in 2011 by WHO. Why I'm showing this? Because this gave us an opportunity to go back, identify those subjects, and try to see if we go for boosting, whether they need any boosting to see the fibrocidal titer or not. So we took this opportunity and identified those vaccinees or the received placebos in 2006 and tried to go for boosting with the same killed oral cholera vaccine. So this was the three groups. One is boosting group, who previously immunized, means they were immunized during the initial trial of 2006 to 9. And then we go for previously placebo primary series. And also we use the placebo vaccinees as no intervention. And they were selected in such a way because we followed them up since their vaccination, they should not have cholera during this entire period. Clinical cholera, that I mean to say. And then what we did on the 0 day after recruitment, we gave them vaccine in the previously immunized group. It is not what kind of previously immunized group. This is called dose 1. And then the primary series who received the placebo also received the dose 1. And we didn't make any intervention in the third group. And we collected blood sample, and then we did the fibrocidal assay, standard assays, and for all three groups. Day 14, also blood samples collected. They received the dose 2, and also this primary series received the dose 2, and no intervention. These are all from Kolkata volunteers. And day 28, we took the third bleed and went for fibrocidal titer for all three different groups. So before I going into the raw data of that study, so what we considered that fourfold or greater rise of serum anti-O1 fibrocidal antibody titer following the second dose. And that was our primary endpoint of the trial, and comparison of the primary endpoint, fibrocidal seroconversion in boosting versus primary series. So we are looking at the two arms. This was done after 5 years of the trial. So initially recruitment age was 1 year and above. So whatever the subjects we received is 6 years and above during this period. And then I tried to see it in a different age fractions. We have a baseline data of day 0. We have data for day 14, and we'll be having the data for day 28. Of course, this is a little bit busy slide. I'll try to explain slowly. We have a 184 number of boosting, 186, and no intervention, 34. It is 80% powered study. And so statistically, we can do all calculations. What we see that in boosting group, the baseline GMT, geometric mean titer of fibrocidal is 137. And also primary series, which means they are placebo group, they are a lesser baseline titer of 94, and no intervention. Basically, these two groups are same, but it varies. But therefore, it is not significant at all in this time. But after boosting means first dose of vaccine and looking at 14 days from the dose 1, we could see that there is a significant rise of the titer from baseline to this. So what happened with the other group who didn't receive any vaccine initial stage, but received the placebo, the ROI also increased. But this increase is lesser than the group who received the initial vaccines 5 years back. And difference is actually statistically significant. And then also we consider the seroconversion here. And then when you go to the 28 days, the same group, the GMT value drops. So what happens when you both give the first vaccine dose, and then look into the after second vaccine dose, we should expect there should be much rise of GMT, but in case of people from cholera endemic areas, it happens just opposite way, it decreased. So Dr. Kadri's group from ICDDRB and also other people, when you looked at their gut homing cells, so the similar kind of situation comes with the volunteers and all. So if we looked into the age 6 to 14 years, the similar analysis, here is also there are increase, and then also primary series is the lesser than the boosting series. If you go to the third group, age 15 and above, then we are trying to see some different changes. Changes are, if you look at that, increase is not that much here, as we have seen in the younger people. And in the primary series also, they were initially placebo, received the vaccine, first vaccine, they are almost comparable. So in adult, in the endemic regions, they are actually, although it's not a clinical defined as a clinical cholera, they're getting exposure. So when I was talking to Andrew regarding the serosurvey and other thing, then also we thought about this. The clinical cholera is giving a signature, is a known signature, is a certificate, but exposure is not as a certificate. So only serological responses can give us idea of their status, whether they are infected or not. And then day 28 also the similar situation. So this is the same data, but in a different format. What we tried to look at it, baseline titer less than 160, all groups, and baseline fibrocidal titer for greater than 160, and tried to look at boosting versus primary, and we really didn't see much here until unless we look into this data. So boosting is showing that 20.7, so it's a GMF rise. And then in the baseline vibrocidal titer from primary is here. So in case of boosting, there are more increase, although statistically not significant, but there are more increase. And in terms of sero response, which we defined as a greater than 4-fold rise from the baseline, it is saying that the boosting is 66 versus 69%. And also, it's for all, sorry. And for when you look into the two different group, less than 160 baseline vibrocidal titer, and the other group with greater than 160. So it is also in case of boosting, it is a little more. To end and to conclude from this study, the study demonstrated induction of comparable level of vibrocidal titer among boost and the primary series volunteers residing in endemic regions. These findings have particular relevance for endemic countries for which long-standing protection is aided by natural boosting via regular environmental exposure. Induction of vibrocidal titer upon boosting will help in maintaining protection in cholera even in the endemic region. The findings put evidences to support that use of OCV boosting regime even after 5 years following the primary series with consideration of shorter interval for the children because of age 5, because they are the lower protective efficacy of 42% on against the 65% protection in average. And this is the data from a single study. I don't want to put a lot of data here. And before I conclude, I'd like to acknowledge we have many people worked for this study. I was also a part of the member of the study. So it's a collaborative study from our institute, as Siman said, and primarily support from International Vaccine Institute, Korea. And with their support from IVI, we also develop our capacity to all these vibrocidal other immunoassays in a standardized form in our institute. Thank you all.
Thank you very much. And again, a reminder to hold questions till the end. Our next presenter is Dr. Gerald Mbua, who is unfortunately unable to be here today. And so we will be viewing his pre-recorded talk on the Africa Pathogen Genomics Initiative and building capacity for cholera genomic surveillance in Africa.
Hello everyone, my name is Gerald Mbua. I work in Africa Centers for Disease Controls and Prevention based in Addis Ababa, Ethiopia, and the Division of Laboratory Systems and Networks. So I'm presenting today about what Africa PGI is, that is Africa Pathogen Genomics Initiative under the Africa CDC, and how it is building capacity for cholera genomics surveillance in Africa. Thank you very much once again for inviting me to present during this scientific conference. So Africa, a brief introduction about Africa CDC and Africa Pathogen Genomics Initiative. Under Africa CDC, which is a department under the Africa Union Commission based in Addis Ababa. The Africa Centers for Disease Control and Prevention was set up in 2017. Under the Charter of the African Union. And shortly after that, it worked with different entities to try to establish itself. And one of them was the African Society for Laboratory Medicine, which is also based in Addis Ababa, Ethiopia. So after the founding of the Africa CDC in 2018, there was what we call the Continental Assessment of Pathogen Genomics and Bioinformatics Capacity in Africa. And that work was also later published in 2019 in the Lancet that addressed the gaps in pathogen genomics in Africa. But again, in 2019, after 2 years after the foundation of the Africa CDC, Africa CDC formed what we call the Institute of Pathogen Genomics under the lab division. And just shortly after that, we had the onset of the pandemic, that's COVID-19. So you realize that the Institute for Pathogen Genomics was baptized by fire, if I may use those words, because shortly after its formation in November 2019, we had the outbreak of the COVID-19, and just a few months later. So it was mandated to lead the sequencing and also the testing under the Africa CDC. And it had a lot of work that it had to do to ensure that the countries that were all under the African Union had the capacity to test and also sequence their own COVID samples. So shortly after that, also in October 2020, that is, we realized that we had to gather all the efforts that we could to ensure that we also establish capacity for sequencing in the continent. Just a few months after we had helped countries establish capacity for PCR testing of COVID-19. So in October 2020, we formed what is called the Africa Pathogen Genomics Initiative, which is also abbreviated as Africa PGI. And since then, it has built capacity for sequencing of SARS-CoV on the continent. But moving forward beyond COVID-19, we are also moving into sequence other pathogens. So a brief look at the Institute of Pathogen Genomics' mandate. The vision was to ensure integration of pathogen genomics and bioinformatics into public health disease surveillance in the continent, and the aim of that to ensure that that is achieved. There are 5 cross-cutting strategic priorities that had to be addressed, as you can see them. And the end of that will ensure that we had rapid and timely response to infectious disease threats in Africa. So Africa is known to be what I can call the continent that has the greatest burden of infectious diseases globally. As you can see this picture, it shows you the timelines of some of the major epidemics on the continent that have come up, and many of them have managed to establish themselves as endemic. Many of these still continue to inflict to take lives on the continent, but also cause morbidity on the continent. So you clearly see that the Institute for Pathogen Genomics had a lot of work to do after its foundation in November 2019. And like I've said before, we started with COVID-19 and we have moved into other infectious diseases on the continent. And as you can see, cholera has been one of the priority areas on the continent, and the Africa CDC, as we shall study, it is also ranked top, and it's number 3 on the continent among the priority pathogens for elimination and surveillance. So the Africa PGI and the Institute for Pathogen Genomics are working to establish what we call the pathogen genomics ecosystem on the continent that will lead to public health decision making using genomics data. So in achieving this, the ecosystem involves establishing or improving capacities of laboratories to test, and also capacity of the sample collection approaches, and also strategies, as we know, and also building capacity for bioinformatics and data analysis. And you clearly see that in each of these areas, the Africa Institute for Pathogen Genomics has tried to do a good job on that, ensuring building capacity for specimen collection, building capacity for testing, and also sequencing, also training, workforce development, and also ensuring there is capacity for bioinformatics and data analysis in the continent. So as I've already indicated, the vision of Africa Institute of Pathogen Genomics, Africa CDC Institute of Pathogen Genomics, and also the PGI, Africa PGI, is to ensure that each of these areas we have capacity to improve each of them, One of them is to ensure that we will provide leadership in working with the member states to ensure that we design impactful pathogen genomics use cases on the continent, and we have seen cholera is one of them, and polio, measles, and so many others. And also build capacity for specimen collection and metadata collection, working with different partners, including the FEJ, based in the University of Western Cape in South Africa. And also ensuring we build capacity for laboratory processing. In doing that, we have distributed a number of equipment, including real-time PCR machines, sequencing platform, and NGS platform, that's from ONT and Illumina. But also we have gone ahead to distribute the robotics for pipetting and also helping with library preparation during sequencing. I've talked about sequencing, distributing a number of platforms. You have trained a number of individuals on the continent in bioinformatics and data analysis. And all that is to ensure that you build capacity for using pathogen genomics to inform public health action on the continent. Yeah, but we have seen like COVID-19 has tried to somehow undermine the so many achievements for many member states in Africa in fighting the endemic infections. So looking at cholera, which is the subject of our presentation today, cholera remains a serious public health concern in Africa, and globally, about 4 million cases are reported and over 143,000 deaths are reported due to cholera annually. And what I can say is that at least 54% of those are from Africa. And from the Africa CDC's pathogen genomic, the pathogen priority list, cholera ranks number 3 as of this year. It is largely, though, eliminated from the industrial countries through treatment of domestic water and sewage. However, in Africa, it remains a serious public health problem. So deployment of the cholera oral vaccine is one of the approaches to ensure we prevent and control these cholera outbreaks. And this is being undertaken at a massive scale as working with World Health Organization. However, sequencing of the Vibrio cholerae is now providing insights into where these strains emerge and spread eventually. This map shows you the burden of cholera in Africa, and you clearly see the first part of that under A, the countries that all regions that have annual incidence of greater than 1 case per 1,000 people. And then B, you have annual incidence of greater than 1 case per 10,000 people. And then for part C, that is greater than 1 case per 100,000 people. So you clearly see that it remains a burden. Even when you try to talk about the annual incidence per 1,000 people, you can see some parts of the continent have a very high frequency of these outbreaks on the continent. So like I said, it remains a big problem. So how have we addressed this? We formulated what we call the Cholera Genomics Consortium in Africa, or CORGEN, in trying to respond to this endemic infection on the continent. So this is formed and it is already constituted, so it has started working, and it is constituted by different players and partners. Here we have the Bill and Melinda Gates Foundation, and then we have the Africa CDC at the center of that, and then we have the Johns Hopkins from Maryland. So these institutions are working to establish this consortium so that we can address 3 core objectives, as we will see in the next few slides. So this consortium has already mapped out countries where the pilot project is going to be deployed. The countries include Nigeria, DRC, Uganda, Mozambique, and Cameroon. And this was, they were selected based on the recent average cholera cases reported per year for these countries. And we are set to sequence at least a proportion of those cases. Some of these samples are already archived in their national public health institutions, and we have already engaged with these countries. In total, at the end of this project, we think we will be able to sequence at least 3,000 samples. This project is set to start on 1st December 2022, and it will end on 1st December 2025, and it's funded by the Gates Foundation and a period of 36 months. So this is the workflow of the project itself. We have already identified this. We have already identified the sites, mapped and also met. We have carried out what we call stakeholder engagement and meeting different players and also representatives of these institutes. So for Nigeria, Cameroon, Uganda, DRC, and Mozambique, you clearly see this belt has cholera as one of the endemic infections, as we have seen from the previous maps. So we will be using archived samples and even prospective collected samples, as we have seen, Korea is endemic in these 5 countries selected. And then we'll be able to perform laboratory testing to identify the toxigenic Korea cell groups, that is the O1 and O1 39, sorry about that. So we'll be retrieving these samples for the countries where these samples have already been collected and then try to go back in time and identify priority gaps where we can select individual samples for sequencing. And then for prospective samples, we will still be collecting them and ensuring that we have enough clinical and epidemiological data as we intend to identify some gaps in the transmission itself. So the labs are already set to start, as we have indicated. This project will start on 1st December 2022. So the objectives of this project, one is to leverage the stored embryo isolates to build sustainable capacity for implementation of cholera genomic epidemiology in Africa, but also we'll be able to identify gaps and strengthen existing cholera testing, sequencing, and data management capacity for local cholera genomic surveillance. And the third objective will be to ensure we enhance skills and knowledge about cholera, about how cholera spreads between outbreaks and within and between countries. So we work with partners, and the outcome of this will ensure that we have a sustainable, or train a sustainable workforce on the continent that can participate in routine cholera genomic surveillance to ensure that in the future, if we are rolling out massive vaccine programs, we're able to have enough capacity to identify the hotspots, but also perform surveillance locally. This is the extract of the monitoring, the evaluation of the program. As we have seen, we have different players in this and different components of this M&E, what they call the training capacity building, sampling scheme, data sharing, analysis and modeling, and also testing and sequencing. And each of these will have a different, or one of these partners taking a lead role. As we have seen, the role of Africa CDC is clearly defined, and also the one for Johns Hopkins University is clearly defined, but also the national institutes, public health institutes in each of these countries will be able to participate, as you can clearly see. So the question always comes up, how do we communicate with these different national public health institutions? Africa CDC has what the Africa PGI has, what we call the help desk, where each of these countries always reach out in case they have inquiries on training, workforce development, sequencing, sample referral, reagents, webinars, data, bioinformatics, and other genomics use cases, as well as potential collaboration. So we always work with these countries to ensure that we address each of these needs. So thank you very much. And I would like to thank our funders who have been part of this journey, trying to develop the concept and the proposal to a stage where we are set to start the first week of December. Thank you very much, and back to you.
It really looks like Gerald is there. For our last talk of the session, we have Dr. Kirsten Wien presenting on Towards Estimating True Cholera Burden, a systematic review and meta-analysis of Vibrio cholerae positivity.
All right. Hi, everyone. And thank you to the organizers for inviting me to share some of this work with you today. So this is a project that I started while I was still a postdoc at Johns Hopkins. And so I wanted to start the talk just by acknowledging the team that I worked with to carry this project out. So particularly Han-Meng Xu and Dr. Andrew Asman at Hopkins, who were involved kind of throughout this project, Dr. John Mwamba, who's in Zambia, who provided us with some data, as well as Drs. Ferdowsi Kadri, Godfrey Bouiri, and Morgan Dominguez, who gave really important insights in how we might interpret the results, as well as some of the policy implications. And so why are we interested in moving towards better or improved cholera burden estimates? As I'm sure many of you here know, the current cholera burden estimates are really important because they can help us to allocate distribution of scarce cholera control and prevention resources, including vaccines. So whether those are estimates like the ones shown on this map here at the national level for different countries or subnational burden estimates, if we have a better understanding of true incidence, we can more efficiently use some of these oral cholera vaccines. But one of the challenges in understanding true cholera burden is that our current estimates rely on counts of medically attended suspected cholera cases. And this is something that Dr. Sonia Hegde touched on earlier in her talk as well. And so if we look at the cholera surveillance pyramid here, we know that there are a number of different pathogens that may cause symptoms related to acute watery diarrhea in the population, and a portion of those will seek care. And so those are the ones that we are capturing in our current estimates of cholera burden. And so we don't know how many we might be missing because they don't seek care either due to mild symptoms or other barriers to healthcare access. A second challenge, which is shown on the top of this pyramid here, is that we're really looking at suspected cholera cases, which will likely be very sensitive to detecting cholera, but not highly specific. And so we can determine whether or not these suspected cases are true cholera cases by collecting stool specimen, testing them. But this is not always used, particularly during outbreaks when resources may be overwhelmed. And so this is really the challenge that we wanted to address in this study. And so specifically, our objective was to estimate the proportion of suspected cholera cases that represent true Vibrio cholera O1 and O139 infections. And so we're looking at O1 and O139 because these are the serogroups that cause the current epidemics worldwide. And so throughout the talk, I'm going to refer to this as Vibrio cholerae positivity or just cholera positivity. And importantly, we wanted to do this across studies from a variety of different geographical and epidemiological contexts, because we know there's going to be a lot of variation in positivity rates. And so a secondary objective of this study was to actually look at factors that might explain some of the variability across sites. And so to do this, we started out with a systematic review of research and surveillance studies that were completed during 2000 to 2001, where in the study area, they had tested at least 10 suspected cholera cases for Vibrio cholerae O1, O139, using one of or more of 3 different diagnostic tests. So either stool culture, which is shown in that picture on the top, a rapid diagnostic test, one of which is shown in that picture on the bottom, or PCR, which is not shown here. And so in each of these studies, we're going to get the proportion positive by each of these different tests. But another thing we know is that none of these tests are perfect. And so in a subset of these studies, and 4 of them, they actually reported cholera positivity results for each different test. And so a second thing that we were able to do was actually estimate how well these tests were doing at actually identifying cholera. And so we pooled results across those studies using a latent class meta-analysis, which allowed us to assume that each of those different tests was imperfect. And I'm going to come back to that in a couple of slides. And then finally, we used the data and estimates from 1 and 2 to estimate underlying Vibrio cholera positivity across all the studies using a random effects meta-analysis and adjusting for test performance as well as study methods. And so in total, after applying our inclusion criteria, we identified 113 published studies from 28 different countries to include in our analysis. And these maps here are just showing that these studies come from a variety of different locations around the world. And so in these maps, the yellow or orange part of the scale shows areas that have higher numbers of studies represented in the data set. And you can see here that they span the years 1992 all the way through 2019. So the data isn't showing up on this figure, but you can see the median estimates from these box plots. And so what this figure is showing is in just the raw data, how does cholera positivity look? And we found that there were a few different characteristics that really vary between where the studies varied in positivity. And so one of those was the diagnostic test type. We found that just looking at the unadjusted raw data, studies that used culture-based methods had the lowest cholera positivity, whereas studies that used rapid diagnostic tests on average had higher positivity. When looking at sampling methods, we found that studies that had higher quality sampling methods, so they systematically tested suspected cases for Vibrio cholera, or they randomly selected samples, those tended to have lower positivity compared to studies that use, say, convenient sampling methods. In panel C, we found that whether there was like age minimum in the suspected case definition also seems to affect positivity. And so specifically, we found that if there was a lower age minimum in the case definition, we had found lower positivity compared to having a higher age minimum, so ages 2 or 5. So setting that higher age minimum in the case definition may help increase specificity for cholera. And then finally, we found that studies that were initiated in response to an outbreak also tended to have higher positivity compared to studies that were part of just regular routine surveillance. And so these are all different variables that we wanted to take into account when we ultimately estimate underlying cholera positivity. But as I mentioned earlier, adjusting for diagnostic test performance is a little bit tricky because none of these tests are perfect. And so without going into the details of the methods, I'm just going to give you a summary of what we found. And so we estimated sensitivity and specificity for these tests using data from 4 studies in Kenya, South Sudan, Zambia, and Bangladesh. And we were able to create a distribution of sensitivity and specificity for each test rather than just a single estimate. And that's shown in these graphs on the left. And overall, we found that rapid diagnostic tests have higher sensitivity, but lower specificity compared to culture and PCR. And so the next step was then to incorporate this into our overall positivity estimates. And so what does that look like? So for reference here in this graph, I'm showing you unadjusted cholera positivity in gray, which is about 42%. But then if we incorporate some of these different things, like diagnostic tests, and adjust for those as well, you can see that we have about 10% higher positivity, so about 47, 48%. And then if we additionally adjust for things like study methods, whether or not we're in an outbreak situation, or a variety of different sensitivity analyses, you can see the results are pretty consistent. And across all of these studies, on average, about half of suspected cholera cases represent true Vibrio cholera infections. But something else that was really important that we saw in the data was that there was really a wide variation in cholera positivity estimates across studies. And so these forest plots here are very busy, and so you don't need to look at all of these details. But this is just to highlight that, so these black dots here show adjusted cholera positivity for each individual study. And so you can see here that these individual studies can range from anywhere from almost 0% positive to almost 100% positive, depending on the country, the study setting, methods, as well as a variety of other factors. And so when we look at the I-squared estimates, they're almost 100%. So there's much more between study variation here than within study variation. And so we also wanted to ask what might be contributing to that variation. And so as I mentioned earlier, there were a couple different variables that we could look at. We found that low sampling quality was associated with higher positivity on average, but in our model, this wasn't almost, but not quite statistically significant. We did find that having a higher minimum age in the case definition was associated with a significantly higher probability of a suspected case being positive. And then being in an outbreak scenario compared to just routine surveillance was about 64% probability higher of having a suspected case be positive. So all of these variables might be contributing to some of the variation that we see. But there are also a variety of things that we just weren't able to measure, which is one of the limitations of this study. So things like the phase of the outbreak, whether you're in the early or mid phase of the outbreak, where you might have higher positivity compared to the tail of the outbreak, we just didn't have the temporal resolution to be able to evaluate that. The actual specific ages of suspected cases likely will impact whether or not an individual is more likely to be a true cholera case, but we just didn't have that data available to measure. And then another really important factor is the prevalence of other enteric pathogens in the study area at the time the samples were collected. And so all of these things might contribute to some of that variation, as well as a variety of other factors. And so what are the overall conclusions from this work? So first, these results suggest that burden estimates that are based on suspected cases alone may overestimate the incidence of medically attended cholera about twofold on average. But despite that, given the high variability that we found between settings, we also can't just simply divide suspected cholera cases by 2 in all settings. So it'll be really important to either systematically test suspected cases using one of these diagnostic tests on the ground, and when that's not possible, to use locally derived data to make assumptions about whether or not positivity should be higher or lower than 50%. And then finally, as far as next steps, as I mentioned earlier, we're only really addressing the one kind of challenge in cholera burden estimation here, which is potentially overcounting cholera cases due to this non-specific case definition. But a really important next step will be additionally to estimate how many cases we might be missing due to things like care seeking or poor clinical surveillance. And so this is something that could be done through a systematic review and meta-analysis of studies that have already been published on care-seeking behavior, or through primary studies, such as the one that Dr. Hegde talked about earlier, where you specifically go and either ask people about care-seeking or do active case finding. And so that might actually account for some of this bias that we're seeing in overcounting, with some of this bias in undercounting, but that remains to be seen. And so with that, I will thank you all for sticking around till the end of the session. If you're interested in more of the details of our methods, there is a preprint with this study available, and it also has links to the data and the code that was used for analysis. So you should be able to scan that there, and that'll take you to the preprint. So thank you all, and I'm looking forward to the discussion.
I'm going to kick off the questions. Willing to offer opportunities to anybody that would like to ask questions of the first couple of speakers. Do we have an extra microphone that we can use for people that are? Bhavtosh, you have a question. Perhaps you can come up here and they'll get a microphone to you.
So my question to Sania, so it was a very nice talk. So Sania, actually, when we do this microbiome study, but we observe that even the normal people who is living in the slum area, they do have also a lot of Vibrio cholerae. So when you did the serosurvey assay, did you find or do you differentiate the infectious versus non-infectious peoples with the serosurvey?
Thank you for your question. No, so when we collected some survey data, when we collected the serum samples to ask whether a person had diarrheal symptoms, but we really weren't able to determine whether the person had, so even if the titer was high, whether or not the person had symptoms. Does that answer your question?
Yeah. And the second question and last question. So with this sero survey, can you differentiate the O1 versus non-O1, non-O1 thinner infections? You can differentiate.
Yeah.
Okay, thank you.
Thank you. Are there other questions? David?
Okay. Also on the seroepidemiology study, I'm sort of amazed at the high proportion of infection, case to infection ratio. And it's hard for me to believe that that data is actually true because the ratio is so high. I'm just wondering, how can you, what is your control group? What can you do to convince me that that data is true? Could you do a similar study in an area that is known not to have cholera and see whether you can really have a control that would convince me that that case infection ratio is as high as you found?
Yes, that would be nice to do, to see whether the case to infection ratio is lower. But I think we're still also working through our estimation methods and trying to incorporate these other pieces of data too. And so this is really just on, it's kind of like a preliminary initial estimate of the ratio. I guess, yes, but that would be, and I think because it is an endemic setting, other studies have also shown that there's much infection in the population. But I think a big question too is what is actually like just exposure versus an actual infection.
With Sonia, because I have questions for her. Okay, so while we are waiting for all of you to remember what the talks were about and come up with your questions, Sonia, I thought I'll follow up on David's question, and I have a couple of questions for you. The first one kind of leads off from where he is. If I understood correctly, if you had a population of 100,000 and you followed them for about 6 months, 40% of them would have some serological evidence that they had been infected and made a response. Forget the exposure, they made a response. Of that 40,000 individuals that we have now that were infected in the last 6 months, only about 80 are going to present to clinical care. That's correct, right? So that if I see figures like this, it makes me think, do we really need a vaccine or not? So I think it makes sense then to think about age stratification and to think about, do you see a difference? Did you look at that? Are you planning to look at that?
That's a great point. Yes, we are planning to look at that. And so actually, Dr. Wayne's over there, Kirsten is working on the same study too, and trying to basically develop an age stratified model to use those 3 components of data that we were talking about, so that clinical surveillance, the healthcare seeking, and the serology. So I think with that, we'll be able to better understand how that differs by age group. But already just from some preliminary stuff, there is quite a difference, at least between the under 5 and over 5 population.
Okay, so my second question, 8 assays is a lot. And you did say this study couldn't be done in other places easily, but were there some assays that were more informative than others?
Yes, and actually that is in Dr. Andrew Asman's work with the random forest model. But the one with the 8 assays was the best performing one. But I think something that we'd like to look at is whether we can get similar results with just using the vibrocidal and CTB ELISA.
Okay, thank you.
Does that answer?
Yes,
Okay.
it does. Thanks. So, okay, while we are waiting, I have questions for Amanda as well. Oh, please go ahead.
I have a question for Dr. So the vibrocidal effect that you have shown, that is for 28 days.
Both 14 and 28 days.
And thereafter, whether it was studied, or I just would like to know about that, and also the immune parameters, if they have been studied or not.
So studies actually were done on 0 day versus 14 and 28 days.
Okay.
And this is only serum vibrocidal titer, no other test, because they are trying to compare the initial vaccination protocol and the boosting protocol.
And how long this data is means
And vibrocidal.
a longitudinal study was done, or?
No, these are the specific set of volunteers who was vaccinated 5 years back. And then we followed back them. We have divided them into primarily two groups who received the vaccine, another group who received the placebo. And then we set up a selection criteria, that elimination criteria. They should not have any known cholera infection for last 5 years. Okay, so these were the setting up this thing. And then we just looked into the vibrocidal titer. No other immune tests were done from the blood sample.
Okay, okay. Thank you, sir.
And your last question is actually, if we looked into this vibrocidal titer, if we go for longitudinal follow-up, and it will definitely went up and it will come down. But in case of people from endemic regions, after vaccination or clinical infection, vibrocidal titer goes up, but it will not come down to the same baseline. Okay, so if you start with the 160, and then after 1 year, it will remain close to 160, but a little bit higher.
Okay.
So Andrews, actually, the forest model is working on that. It is a kind of a 1-point study in a different time points and looking into predicting the infection past 1 year.
Thank you.
So my questions for Amanda, you were talking about, this kind of relates to what Gerard also said, that the African PGI initiative is looking at sampling different countries differently, higher proportions in countries that have fewer samples, lower proportions in those with higher samples. But in your presentation, you talked about different countries and what were the strains that were circulating in them. And I just wondered how generalizable that was. Most often, epi studies tend to be done in places where the disease is. And if you have new areas emerge, were those oversampled, or was it even possible to obtain samples from areas where disease was newly emerging?
So if I understand your question correctly, in our studies, you're asking if we were able to obtain samples not just from the height of outbreaks, but from other lesser, smaller outbreaks.
Or newer outbreaks.
Or newer outbreaks, yes. So I guess what I would say is it depends on the setting. If we were conducting surveillance in advance, or whether we were trying to do an outbreak response, like in Kenya, those were outbreak response samples. And Tanzania was the same. Malawi was samples that had been stored in their repository. But in Cameroon, we'd been doing surveillance, and in fact, didn't find very much cholera. So those would have been newer outbreaks. So I think it depends on each setting. And as it pertains to the AFCDC, Dr. Sack may be able to answer more. Those countries are completely separate, but they were also picked specifically because of their cholera burden. And for example, Mozambique is a little out in left field in comparison to the other countries, but as Jerome pointed out, it had one of the largest cholera outbreaks to date, and it seems to spread often into Malawi. So we hope in that grant to set up more surveillance sort of as I described, so that we aren't just getting the outbreak response samples and we're able to get a little bit more of the gaps filled in. Does that answer your question?
Okay. Andrew has a comment, question.
Hi, thank you. So it's for you / Africa CDC, so you can. So it's exciting to see what's going to happen over the next years with more sequencing in Africa. I guess one question that I have is thinking about what you talked a lot about new introductions and lineages and how we think that may relate to risk of outbreaks, and is a new lineage indicative of any way of increased risks or a larger outbreak, or is there any clinical meaning to it? And if we don't know, how we might answer that with some of the sequencing that's going to be done over the next years.
Okay, so let me recap again. So you're saying, are we going to be able to collect the metadata, for example, that is associated with each sample to understand the risk with each new introduction?
Well, I mean, this focus on introductions, it's very interesting to understand how things circulate. But is there a clinical meaning? Is there something that's relevant in terms of public health to saying we've identified a new introduction? I think there's many reasons why it help us understand cholera in general. But does a new introduction mean, like, should the country be worried when there's a new introduction?
Right. Yeah. I think you probably have answers to that as well. I think it's kind of interesting. Actually, in a recent discussion, someone made a comment about how Kenya doesn't have endemic cholera, it's just introduced. So they weren't as concerned about vaccinations there, right? So I think it is about the approach that we use. If we think it's about introductions and then it's eliminated and then there's no cholera until someone else brings it back, right? So I think that is where we might need to change our perspective about what an introduction means. How do we prepare for that risk in advance so that we can prevent that cycle? And I don't know how these specific countries will relate to that. You have more experience in DRC than I do. The countries I was looking at in Southeast Africa aren't really involved. But DRC and Nigeria, we consider to be more endemic, whereas Cameroon might be more that introduction. So I think you posed an excellent question. David, do you have anything that you wanted to add?
I was very impressed with Dr. Nandi's talk. And my question is, and I think other studies have seen the same thing, that, and you alluded to this, that when you give the first dose of vaccine, you get a good response. When you give a second dose, you think that you would get a higher response, but you don't. In fact, it's lower. And I think other studies have found the same answer. And I'm wondering how you interpret that. Why doesn't the second dose give you a higher response?
So it's a good question. And what we believe that this happens from in the volunteers from the endemic regions, but it's not happening for the volunteers on the developed countries, where we could see the higher response after the second dose. What we believe that is a pre-existing immunity. Once we boost it, is the first dose. What we say first dose is basically the boosting, because it's already pre-infected, already having some immunity to it. And then the second dose is actually the very close, the second dose. So it will not induce more immune responses, rather it will be snubbed. So that was our hypothesis. Yes, the baseline titer is there, but when we think of the vibrocidal only, and we looked into the vibrocidal titer, it's a baseline titer, we called it, but it won't give you idea for their pre-exposure, because I'm saying, so for cholera cases, for the vaccine studies, the baseline reaches about 1 year, and then protection continues for 5 years. So in that case, all those memory cells are there when it is exposed. So it will be a quick response from those memory cells. So first dose maybe for the vaccinees from the endemic regions is actually acting as a kind of a boosting, that we believe. And the second dose quickly within the 2 weeks also is a second boosting. So then it's not the expected titer from the top to bottom.
I have a question that if you compare between the endemic and the non-endemic pre-immune serum, so do you get a difference between this? Okay.
Even in India, when we did some early studies for the cholera vaccine, not with the Sancol, and it's a vaccine from Dr. Amit Ghosh and our group. It's a live VA 1.3. And then we did some studies with Dr. Mahalanobis. Actually, I'm fortunate I worked with Dr. Mahalanobis in ID Hospital for these vaccine studies. And with the Kashmiri volunteers from India, there is a non-cholera region. When you see the baseline titers are very low. And when we use this vaccine for this very limited number of Kashmiri volunteers, we didn't see after the second dose that this kind of scenario. I think I answered your what? Thank you.
Okay, I had one more question for Amanda, which was around the cool tools, which is if you've got filter paper and you've got RDTs and the RDTs can be reused, you spoke about bacterial pathogens, and within the bacterial pathogens, is there a variability in the amount that you can recover even for sequencing? So for example, you would expect there to be, if you had an RDT on a patient specimen, to see more Vibrio cholerae spotted onto filter paper or applied onto the RDT than say Shigella. So when you try to sequence, do you get better recovery for, let's say, Vibrio than Shigella?
Yeah, I think that's one of the things we're working on. We have shown papers that if you put the whole stool and some of the suspect cholera's case that test negative by RDT, you can detect Shigella and others. And we haven't yet tried to sequence those, but in the case of where we see a co-infection, we're working on that with the metagenomics to see how much we can assemble. But I think that there's a lot still to find from that. But I am starting a new grant with Gates on adult diarrhea based on filter paper. So I'll tell you more soon.
So are there any other questions?
So this is a question for both, I guess, Amanda and Kirsten. One of the questions is, it's exciting the work that you're doing with the filter paper because it really broadens the information that we can get on lineages. With the whole genome sequencing, are you looking at fibriophages as well? And the question for Kirsten is, I know you looked at a number of things that affected the positivity rate in rapid diagnostic tests, and did you include the prevalence of fibriophages as well and their impact on false negatives in the rapid diagnostic tests?
So phage, however you say it, is involved in our grant. And I just emailed the preliminary metagenomic data to Eric Nelson. Our colleagues at Baylor are very interested in it as well. If you saw the one sample that was completely ICP1 with very little Vibrio cholera, that wasn't supposed to have been an isolate, actually. So I think that samples like that are really interesting to see how much of it actually proves to be ICP1. So I think there's a lot to investigate there. So hopefully we can figure that out soon.
Yeah, thanks for that question. And the short answer is no, we just didn't have that information available to adjust in our analyses, but like definitely that's probably contributing to some of the variation we see.
So if there are no other questions, I'd like to thank all the speakers for doing a truly outstanding job and the audience for the very engaging questions, and back to the organizers now.
Thank you, ma'am. Thank you both to the chairs, Dr. Kang and Dr. Debis. I request you, madam, to kindly felicitate our speakers of this session, Dr. Debis and Dr. Kang both. So please. We are delighted to have Dr. David Sack here, and I will request sir to felicitate the chairpersons, Dr. Kang and yeah, please. जाओ, जाओ. हाँ, चले जाओ. Dr. Davis. हाथे खुले दो. Yeah, I will call Dr. Hedge. Dr. Hedge. Dr. Sonia. Dr. Nondi. And Dr. Wins. Thank you all. Thank you, Dr. Kang and Dr. Debis. So before we break for lunch, I have an announcement to make that for travel grant assistance, please contact our help desk in the first floor. And I also request all the speakers to please upload their presentation for the next session. Okay, so we will end the first session here, and we will go for lunch, and we will be back here at 4:15 p.m. after the plenary talks in Hall A. Okay, thank you, everybody. Enjoy the lunch.
हेलो चंद्रेश भाई, हेलो हेलो, यहाँ से ऑडियो अनम्यूट किया है ना? हाँ बोलो, चुप बोलो। हेलो 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 हेलो, जीगू भाई, हेलो जीगू भाई। हेलो 1 2 3 4 5 ऑडियो आवे છે? हाँ ના જી છે, 1 મિનિટ મારા અંદર જવું પડશે, 1 મિનિટ મારા. हाँ जुम्मा कनेक्टेड છે, वो बाहर निकल गया भैया, फिर से करो रिकनेक्ट. ये वीडियो खोलो. हेलो चेतन भाई, चेतन भाई,
બોલો બોલો.
हेलो, उसका ऑडियो
हेलो
दो,
अवाज आवे છે?
चेतन का.
हेलो
अच्छा उसका ऑडियो देख लीजिए जाके. हाँ हेलो हेलो
हेलो आवाज आ रहा है
चेतन
क्या?
भाई,
हेलो
चेतन भाई राणा,
हेलो
हेलो हेलो
हेलो
हेलो
बोलो बोलो.
हेलो हेलो, और थोड़ा बढ़ाई दे. हेलो हेलो
हेलो
हेलो हेलो खुला है. हेलो हेलो हेलो, वो साली बैठा है ऐसे.
हेलो
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 चेतन भाई,
हेलो हेलो
चिंतन भाई,
બોલો બોલો.
આવે છે ને અવાજ?
હા મારો અવાજ આવે તમને?
હા આવી ગયો.
હમણાં હું બોલતો હતો પણ તમને મારો અવાજ નતો આવતો.
હા એટલે અહીંથી અનમ્યુટ હતું ને એટલે.
હા ઓકે
બરોબર હા
સારું.
હા ને પછી મારો નંબર છે ને તમારી પાસે?
હે
મારો નંબર છે ને તમારી પાસે કે નથી?
છે ને
એક મિસ કોલ મારી દેજે ને મને.
ભૂમિનભાઈ ને
હા હા જીગુભાઈ થઈ ગઈ વાત. થઈ આવી ગયો. હવ હવ
મિસ કોલ માર્યો.
હા ને તું ગમે શું નામ લખ્યું તારું?
ચિંતન.
હવે વિમુક્તિ વ્યાસ. ઓકે ઓકે આવી ગયો
આવ્યું
આવી
ને
ગયો. હા
સારું.
ખાલી એક સંભાળ लिया
Good afternoon. I'm Anne-Laure Page, and I'm part of the in vitro diagnostics assessment team in the pre-qualification unit at the. What I would like to present to you today is the general overview of the
Check. Hello. Hello. Check. Check. Hello. hello.
Ladies and gentlemen, welcome to parallel session in Hall B. The topic of this session is genomic evolution and diagnostics, enteric pathogens. I call upon stage Dr. Supriya Kumar, Bill and Melinda Gates Foundation, USA, and Dr. Nicholas Thompson, Sanger Institute, Cambridge, UK, to lead this session. Sir, madam, please. I will hand over this session to you. Thank you.
Thank you. My name is Supriya Kumar. I'm a program officer at the Bill and Melinda Gates Foundation. Welcome to you all to this session. I'll be co-moderating this with Nick Thompson. And we'll go pretty quickly through the talks because we are very behind schedule. So the plan is to take just one question after each talk. Each talk will be about 15 minutes long. So let me introduce Nick, and then he will take over and introduce the first talk, and I'll come back to introduce the rest. So Nick Thompson is the head of parasites and the microbes program and group leader of bacterial genomics and the evolution group at the Sanger Institute in the UK, and also professor of bacterial genomics and evolution at the London School of Hygiene and Tropical Medicine. So Nick.
Thank you very much. Thank you everyone for coming. We've got quite a diverse and lively session. Our first speaker is Arif Tanmoy, who's just about to finish a PhD in April of next year, hopefully, and at the Child Health Research Foundation in Bangladesh. I'm going to talk to us about Paratyphi A, about a surveillance framework and tool, so something that's much needed. So Arif, come on, please take the floor.
Hello, everyone. Hello, welcome to this session, as I am the first speaker. Okay, I know you were a bit tired. There are long talks. I don't think I will take the whole 15 minutes, so it will be like at least 3-4 minutes short, I guess. Okay, so I'm Arif Tanmoy. I'm from Child Health Research Foundation in Bangladesh, and for some time now we have been doing working on a project for paratyphi A, which is a very neglected topic these days. In fact, ASCOD is a meeting of enteric diseases, right? I actually searched how many presentations are there in this meeting, and I found 3. So, so it's pretty neglected. We can get from there. Okay, so the topic of my presentation is we have been working on a tool, Paratype. It's a genotyping framework for Salmonella paratyphi A. So since it's a rare topic, I get, so let's get a few brief outline first. So what is paratyphoid? It's a febrile illness caused primarily by Salmonella paratyphi A. It causes more than 3 million episodes every year and almost 20,000 deaths globally. It's endemic in LMIC like typhoid, and it is symptomatically similar to typhoid. And it is so symptomatic similar to typhoid that there is no different treatment regimen for paratyphoid. For paratyphoid, the treatment regimen we use, those are mostly based on the AMR data of typhoid. So we tried to see why is that. Another factor is that for typhoid, we know the typhoid vaccine is coming in many countries. In some countries, we already got the vaccine. In some endemic countries, the vaccine will be there maybe within a few years. But just like I said before, the data, the paratyphoid treatment is based on typhoid. So if typhoid is gone, then what's it's going to be? And are we ready for that? When we looked at that question, we actually found out that paratyphoid is not a neglected infectious disease. It's actually the most neglected infectious disease. This is a paper published in 2019 on PNS. They actually compared Delhi's with the number of publications. And they saw paratyphoid is the highest. Paratyphoid is the disease with highest Delhi's and the lowest publication number. So based on that, we wanted to know why is that? Maybe it's because of the lack of AMR, because for typhoid, we know that like AMR is very, very high for some antibiotics and for some it is low. But for paratyphoid, it's almost nothing there except quinolone, except fluoroquinolone. This is the data you are seeing here. This data is basically a data set of more than 2,000 cases that we have been generated since 1999 at our foundation in Bangladesh. And we saw almost no resistance is there, and there is no MDR. And mind it, this is a data set of 2,000 cases, more than 2,000 cases that we are talking about. So the thing is, we also looked for the genome data. Is there how many genomes are available for paratyphoid? And we saw very limited data. This is a graph for that. This is published in PNS in 2014, and this was back then only 150 isolates, 150 genomes was available back then. And the analysis they did, they differentiated the whole population in 7 different lineages. But the thing is, South Asia had 82%, has 82% of paratyphoid cases globally reported. That percentage was not there in this data set. So the population structure that we saw in 7 different lineages or something like that, it's probably not complete. So few things is clear that we have a lack of data and we do not have a genotyping system. And both of them are needed for surveillance these days, and mostly if we are talking about genomic surveillance. So we tried to solve that problem. At first, what we tried to do, we tried to build a global database of paratyphoid genomes. For that, we had I said earlier that we had a biobank of more than 1,000 isolates. So we sequenced almost half of them. we sequenced 528 new genomes from Bangladesh. And then we gathered another 250 genomes from Nepal and Pakistan with our seed partners. And then from the literature, we found that by then, more than 500 genomes were already there. So we made a global biobank, like global genome data set of paratyphy A of 1,379 isolates, nine genomes, which had covered 37 different countries, and year timeline was 1917 to 2019, 102 years. Then using the database, we did some analysis with the phylogenetic analysis. In fact, this is, when we did this analysis, we thought that let's see how the previous lineage system works. So we added that. And when we did that, we saw there are many gaps because there are so many different clusters now that we could not assign lineage to many isolates. So then we wanted to build a new system then, because we need a new system. Otherwise, we cannot segregate the whole population in different category and different genotypes or lineages, whatever you call it. So for that, we used that previously done phylogenetic tree and used FastBabs to get the clusters, the genomic clusters out of them. And then we randomly selected 315 isolates to do the phylogenetic analysis to see the whole population, to see the whole population evolution of the paratyphy A genomes. And we saw that the whole paratyphy A population till now is segregated in three different primary clades. The one clade you can see, it's only one isolate here. It's an isolate from Hong Kong in 1971. That isolate is so weird that it does not match with anything else. Absolutely nothing. So our conclusion was this is probably like the last member of maybe 100 years before ancestry or something like that. And in the whole tree, you are seeing all the colors. It's all of them are new genotypes that we are calling. And we found total 18 different genotypes in the paratyphy A population and total nine different secondary clades. And the colors you were seeing, each color is a genotype. Once we got the genotypes, we tried to see if it is conserved, like if we can define them with an allele, and we did that. We found we got the allele for each of them on a conserved gene. And using that, we used the previously done phylogenetic tree, and we put all of the definitions there to see if that definition matched the whole data set, because the scheme we got, that was from 315 isolates, but the whole library was more than 13,000 isolates, 1,300 isolates. And we saw like 100% specificity and sensitivity. It matched perfectly to the whole tree of almost 1,400 isolates. Then we used that definition. We wrote a Python script and named it Paratype. It's already been released in GitHub though. So when we have that, we know the paratype is there to genotype paratyphy A genomes, but we wanted to know what is the global genotype distribution and the AMR distribution looks like. So this is what we got when we saw the distribution of different genotypes. We saw many different countries, like the dominant endemic countries have different, each country have different dominant genotype. Like for Bangladesh, it has genotype 2.4.4 dominant here. In Nepal, it was 2.4.3. In Pakistan, it was 2.3.3, which is also similar to a paratyphoid outbreak happened in China in 2011. For India, the data was very minimum, and we saw a mixed of dominant genotype. There was no like absolutely dominant genotype there. And when we looked at the AMR, we saw absolutely no AMR, like only 2% of the isolates of the whole 1,379 genomes had at least one AMR genes. But in case of the QRDR, the fluoroquinolone, almost 85% had at least one QRDR mutations. That means either mutations in GARASE A, GARASE B, or PERC, something like that. So I was saying about that before, that the Paratype tool has been released on GitHub, and it can predict the genotypes of a Paratype A genomes, and along with that, the QRDR mutations is there. It can use the tool, it can use the almost all data formats, like the raw data format from either Illumina or Nanopore platform. It can use the assembled contigs. It can use the mapping file like BAM file or VCF file. The medRxiv paper, it has more details on it, obviously. And this paper, the Paratype paper, has been recently accepted on Nature Communications. So I'll press that. And so now we know the Paratype is here for Paratype A, and we hope that it will help future genomic surveillance studies, and it will help nations to plan interventions and treatment studies. So acknowledgement, there are so many people, especially our SIP partners, KCR, Jason, Joy Dyson helped us a lot with the script. Stephen Baker helped us a lot with the AMR, and also the script was there. Luby and Gordon Dugan was kind of hallmarking us with the research, and many helps with BMGF, obviously, with the funding. And some of the, like at the starting, we didn't have the funding, so we started with our core funding, and then BMGF got with us. It was excellent work in the end.
Thank you very much, Dr. Tannoy. Do we have any questions? We can take one question, and then we'll, yeah, the back there. Do you want to grab? I don't know how this works, actually. If you.
Thank you for the presentation. Really, Paratype is the neglected paratype for disease. As you have shown that the development of AMR is around 2% in Paratype. So is there a logical reason based on your genomic design of the Paratype that is the late development of AMR in these bacterial species only? Because the counterpart typhi already resistance to the third generation cephalosporin and waiting for the carbapenem resistance generation.
Can you speak a bit louder? I can't hear it properly, sorry.
Hello?
Yeah,
fine.
Please.
So my question is that, what is the reason of slow development of antimicrobial resistance in Paratype A? While the counterpart typhi, S-typhi, is already resistant to the third generation cephalosporin, and awaiting or some countries already reported the carbapenem resistance. So is there any logic behind the genomic data of the available Paratype A you have shown in the presentation?
Okay, I think I get the question, but I'm afraid I do not have the correct answer yet. It's a very, very intriguing question, I would say, and I think some of the groups in the world who do the basic studies with Paratype and Typhi, they are actually looking into it. I heard from a couple of groups, but I do not have an answer, the exact answer. But what we know that from a study, I read that Paratype A cell is often it cannot hold on to the INCHI1 plasmid, which is the main plasmid which carries the MDR genes for typhoid. So that can be a reason. I think I heard of like 1 or 2 paper for this. I don't even remember the author's name, so I can't give you the reference, but I read it somewhere. But other than that, I'm not sure like what's the exact reason for this.
Okay, thank
I'm
you.
sorry.
Thank you very much.
You're welcome.
Okay, thank you very much, Dr. Tannoy. Thank you very much.
Thank you, Tanmoy. So from paratyphoid to another neglected disease, though it's one that we've been hearing quite a bit about in this conference, Nick Thompson is going to talk about antibiotic resistance uptake in cholera in Yemen.
Good afternoon, everybody. Thank you very much. I'd like to thank the organizer of the conference for giving me the opportunity to both chair the session and to speak. And thank you very much for attending. My talk is around some recent work we've been doing in the Yemen, and about the sort of phylogenetics of strains within the Yemen, and some of the more phenotypic shifts that we've observed recently. This is work that in my lab was led by Florent Lassalle. Okay, so for many people, this is a very familiar story. So the Yemeni, but just for background, a bit of introduction, the Yemeni outbreak began in 2016 and is the largest cholera outbreak in recorded history. It started following a humanitarian crisis and a war in 2015. There have been well in excess of 2.5 million suspected cases, many of which were in children under 5. And if you look at the epidemic curve, what you notice are clearly peaks and troughs from 2017 or 2016, 2017, right through to 2020. So there's a lot of dynamics within the cases within the Yemen. And what we wanted to do was to bring a genomic view to that dynamics and try and understand the nature of those Vibrio cholera strains causing disease. This was our original study, which is looking at the isolates from 2016, 2017. And there's some complicated pictures on here, but I'll try and explain them. Actually, they haven't come out very well and very clearly on the screen, so I'll try my best. But so this is a phylogenetic tree. It's a bit blurry, so I'm just going to point out the major features, and this is the one we're going to concentrate on. But earlier in the day, Amanda Debs spoke about the pandemic 7 lineage. Actually, the pandemic 7 lineage is the epidemic lineage responsible for almost all cases, not all, but almost all cases of cholera within the current pandemic. And this is a phylogenetic tree with the root. It's a circular radial tree. And whilst you can't see much of the detail, what you can see are these branches that sort of radiate out from that central sort of trunk. And they are the introductions of cholera actually from South Asia, which is actually in green, for strains introduced into Africa. And the T numbers that we came up with at the time stood for transmissions, and they meant transmissions from South Asia into Africa. And at the time we actually published this first study, there were 12, but there have been several more now. And the current one that we're focusing on is T13. And this is actually the more detailed tree of T13 and of T12 and T13, this part of the tree here. And I'll just go through it very briefly. At the root of this part of the tree, we can see South Asian strains from India and Bangladesh. We can see strains here which share a common ancestor with the earlier Indian strains. And actually, honestly, these are highly related clones, so very little variation in them. And these strains then seeded outbreaks in T12 into West Africa. So this is a T12 introduction into West Africa. Between T12 and T13, we have isolates in India and Nepal, and we have the Haitian outbreak, the Haitian isolates, which were associated with the movement of UN peacekeepers from Nepal into Haiti. And then subsequently, we have isolates actually from this region, actually from Calcutta, which actually were isolated during epidemics across India and Bangladesh. And then these strains here at the end are the introduction of strains from South Asia via East Africa, so Tanzania, Uganda, Kenya, and so on, before we got to the Yemeni isolates, which are at the end here. So in summary, the Yemeni isolates were grouped into a single clade, which is this clade at the end here, on the end of a long set of very closely, tightly related branches that generally show movement from South Asia, or all show movement from South Asia into, in this case, Africa or Haiti, or in this case, from South Asia to East Africa to the Yemen. So it was a single epidemic introduction into the Yemen via T13, which was responsible for the 2016-2017 outbreak. And whilst you can't see it because it's behind my chair, it says down there that they were antibiotic sensitive. And actually one of the biggest observations around the early introductions were they were largely pan-sensitive to antibiotics. And this is kind of summarized here, because actually that changed at the end of 2018 and 2019, where isolates were phenotypically seen to be multi-drug resistant. And so and that change coincided with an increased number of cases. And it also, in context to that, in the Yemen, azithromycin, if you remember the number of cases that were in under 5-year-olds, over a quarter, azithromycin was actually given to pregnant women and children, and doxycycline to all others until the end of 2018. So there was a very strong drug regimen. You can see here the actual antimicrobial compounds, and you can see here the sensitivity or resistance profile for the 16, 18, and 2019 isolates. And you can see sensitivity and then resistance, often to some unusual ones like third generation cephalosporins, but also things like macrolide, so azithromycin, that was resistant within the 2019 isolates. So a huge change in drug resistance, a massive switch almost overnight from pan-sensitive to multi-drug resistant. That's a huge change. And given that this is both evolution and diagnostics, this session, that huge change is relevant to both. One, we need to understand how there can be such a huge shift. And two, we need to think about what we're actually trying to diagnose and track, whether it be the lineage or whether it actually be genes that are therapeutically or important for control. So with the National Health Center, the National Center of Public Health Laboratories and Abdullah Al-Harazi and colleagues in the Yemen, in Sana'a province, and the Ministry of Health in the Sana'a, they've collected enormous numbers of samples and actually been using culture to confirm Vibrio cholera 01. So the ones that we sequenced were a subset of these culture confirmed Vibrio cholera 01. And I'm going to focus purely on those. I'm going to put them in context of the Vibrio cholera species, not just now the pandemic 7 lineage. So if we take the whole species, and these trees are actually the same, essentially the same thing, one's flattened out and one's circular. But this gives you an example. So this is the whole Vibrio cholera species, the root here. These isolates here are non-pandemic 7, so non-epidemic lineage, and I'll summarize that in a second. And these are the ones actually that caused the outbreak in the Yemen in the pink. And if we switch to this tree here, what we can actually see is that if we look at all the Yemeni isolates, the pink ones are these ones here, the 7th pandemic L-Tor. There's a small triangle that denotes the pandemic lineage here. The majority of isolates from the Yemen that we sequenced that were culture confirmed for 01 were from this epidemic lineage. And then there's a minority of other strains. These are what we refer to as endemic strains. They cause sporadic disease, but they tend not to cause, and certainly didn't here, cause linked epidemics. And so these are the strains that were dominated during the 2019 epidemic. And I'm actually going to focus on them a bit more. In this case, I'm going to go back to the single lineage. So that was the species, just to show you that there were non-7th pandemic L-Tor isolated during the Yemeni outbreak as well, but they were in the minority. But now if we focus on the epidemic lineage, what we can do is we can divide them in beautiful colors to try and subtype them a little bit. The names, the letters, the numbers don't really matter. Actually, the colors are probably more important for the purpose of this talk. But what I hope you can see, all these isolates here in this tree here, you can see the expansion. This is a ladderized tree. You can see the expansion of isolates along here. These are all the Yemeni strains. And actually there were 3 clades, 1, 2, 3, 3 major clades that explain the dates 2016, 2017. This is the first outbreak. 2018 in brown here is that largely these strains here, and then 2019 here. These are the strains that caused the disease in 2019. And so the key point for us was what happened between these 2 lineages, and they're all very closely related to this lineage here that made that such a phenotypic difference in terms of antimicrobial resistance. And so there's all sorts of things. It could have been a new strain was introduced. It could have been that actually endemic strains within the region had acquired drug resistance in some way, but rather dramatically, or a new strain could have been introduced from outside the region. The first thing you want to understand, if you want to understand, for example, why these are different, if it was to do with a local emergence of drug resistance, you might expect there'd be a strong geographic signal as strains acquired resistance and then spread out from a foci. Down here, you may not be able to see it, but actually, if you take all 3 of these major clades, actually there is no significant spatial segregation in terms of phylogenetic clusters. So they actually occupy almost exactly the same space. If you look at temporally, you can see this lineage here, and the pink lineage. You can see that as we'd predict, these ones here, 2016, 2017, these ones here were 2018 in pink, and these ones here were 2019. So whilst there wasn't a strong geospatial separation, there was a very strong signal in terms of the years and their dominance over different years. So what does that tell us? It tells us that potentially one strain hasn't dominated, become resistant from 2016 and then spread and caused disease in subsequent years. And that's the explanation for the drug resistance. What it does tell us is it could either be an introduction of a new lineage, and that's probably the most likely scenario. So what do you do then? Well, actually what you do is you go back to basics and you say, well, Vibrio cholera has a core genome of about 4,000 genes across 2 chromosomes. And so you ask the question, what's the difference between the 2018 and the 2019? So classic comparative genomics. And in this case here, if these are actually all the pandemic 7 strains, this block of blue means that the vast majority of core genes actually in Vibrio cholera, if you mark them as present or absent, are actually conserved across the pandemic strains and actually beyond. What I hope you can see here very quickly, and this is the power of just of presenting data in this way, is a block here. And bearing in mind at the moment, this is just blue ticks on a slide, but I'm going to show you what that actually means. But this block of blue here is the reason that actually 2019 strains are different from all the others. And it also explains why there was treatment failure with macrolides. So if we take that, because we have the sequences, we can assemble and look at it. And what we see is within that block of genes, there's 140 kb INC plasmid. And that INC plasmid is quite an interesting one because it's self-mobile, so it has mobility genes. It can spread between isolates. So that's pretty important because it means that the impact of acquiring that plasmid can go well beyond actually the isolates that carry it. But it also carries this region here, and this region here was unique to the Yemeni strains. It's a composite transposon, which encodes antimicrobial resistance. So what we had was we had a new strain in 2019, became multi-drug resistant, carries a new plasmid, and that plasmid itself has 2 different natural histories. This is a mobile genetic element. It can jump between the chromosome and different plasmids, and we have a plasmid that's self-transmissible. Actually, one of the plasmid backbone was also seen in previous African isolates, but with a different composite transposon. If we look at that transposon itself, it's pretty scary. We have BLAR-PER7, which is actually quite an unusual extended spectrum beta-lactamase. We have aminoglycoside resistance. We have sulfonamide resistance. QACE is a quaternary ammonium resistance. That's a disinfectant. And then perhaps more interestingly and worryingly, we have the macrolide resistance genes here. So 3 different macrolide resistance, which explains the resistance seen in the multi-drug resistance. And if we take the actual, then we say, well, where did this come from? And so what we did here is we take the plasmid, which is in if it doesn't, it stays white. What you can see here is Klebsiellas, Vibrio, Pseudomonas. So going out, these are Klebsiellas and E. coli. This is another Vibrio sequence actually from Zimbabwe, and this is a Pseudomonas. And actually what we found, of course, is other enteric bacteria have the plasmid, but what they tended to differ, they didn't have the composite transposon. And then conversely, we found other bugs that had the composite transposon present either on the chromosome or a different plasmid. So what it means is the Vibrio cholera is really drawing from a very rich background of other enterics, both in terms of their plasmids, INC-Cs, and in terms of the transposons that they're carrying. And if, as I close off, what you actually see here is the 2019 isolates here in this sort of beautiful color, and you can see they have both the plasmid and the integron. Almost all of them do, so it was a beautiful acquisition. You might notice here an isolate here that was actually in this clade here, the only one isolated actually in 2019, which carries the a single plasmid and the integron, and we think that's overspill from these, so we know they're coming into contact. And we also find these strains up here from Zimbabwe, which carry a similar plasmid with a different integron. So we have outbreaks of 7th pandemic LTOR, which carry INC-C plasmids, which confirm multi-drug resistance and which have been acquired by the 2019 strains and introduced into the Yemen. And I'll just summarize hopefully all the things I've said here. So T13 strain, we actually found environmental strains as well as our T13 causing disease, but very low level. We actually found mixed infections in some people. And we found diversification of the T13 strains, 3 different clones dominated between 2016 and 2019. And then we found this MDR clone, which explained a lot of the drug resistance. Plus we found spillover of the strains from pandemic 7 lineage into actually endemic Vibrio cholera strains, as well as other lineages of pandemic 7, i.e. there's really is a dynamic interaction between both environmental and pandemic strains, but ultimately the pandemic strain caused the epidemic. Thank you very much.
Maybe that's it. Yeah. Just one question for Nick, please. Any questions? Yes, please.
So what is the MLST data? The multi-locus sequence typing, this is 6 or the 555 you mentioned?
Yeah, so the MLST data for those strains, for the Yemen. So all of
endemic versus epidemic strain.
Yeah. So actually, okay. So the epidemic strains, they're all the same MLST type. I think it's got the major environmental clone where we had 21 isolates was ST555. So that was the environmental clone or environmental strain. That's also been found in India. But I think it's about, when we compared the strains that we've seen before in India to Yemen, they were something like 800 SNPs apart, which actually is for cholera or for Vibrio cholerae, that's actually quite a lot of variation between the two. But yeah, they were largely ST555. Yep.
Great, thank you, Nick.
Thank you.
Do you want to introduce the next speaker? Do you want to introduce the next speaker? I can, if you want. Yeah. Our next speaker is Dr. Chandrasekhar Nair, who is the co-founder and CTO of Mall Biodiagnostics based in Goa. You have to come up from that.
Hello and good evening. Thanks especially to the organizers and all the support that we've received from NICED, and just want to start off the presentation. If that comes on, how does it come on? Yeah, that's that. Great. So let me start with a story. It's been a long day, so an interesting story. On the 4th of July, I got a call from Dr. Varsha Sridhar, and she runs a very innovative company in Bangalore, asking whether you have any cholera test. So we've been working on this cholera test, and we were getting this validated here at NICED. And I told her we have a research use only test that has undergone some analytical validation. So she said, we need your platform, we need your test, we need them now. And we were able to load the boot of a car, and she went to Pondicherry where there was a suspected cholera outbreak. And by next day afternoon, they were able to show some results from some samples that were there, environmental and otherwise. So the basic idea was that starting from scratch, to set up something and very quickly get results was possible because we had this TrueNAT platform. And what is this? This is a battery operated, field usable real-time PCR, quantitative real-time PCR system. This has been actually the story of our development has been the story of India's development. And Dr. Ganguly is here, and I have to acknowledge his massive contribution to the early design of this platform, supported by Nimitli, supported by the Department of Biotechnology and by ICMR. So in some sense, this is a platform that I think most of India has actually worked on in various ways. This is battery operated, this is field usable, and we can actually transport this in real time wherever we need to go. One of the reasons why this platform is so versatile is the sample prep. And we kept the sample prep separate right from the very beginning. We started with magnetic nanoparticles that we developed and characterized, and eventually moved to nanofibers. Doing it this way allows us to get very aggressive with the prep as and when we need it. So our primary deployment around the globe today is for MTB, and we were able to process sputum samples and all the extrapulmonary samples with ease with this platform. But so can we do stool, we can do blood, we can do various kinds of samples. I forget to mention that that is also battery operated and gives you total nucleic acids extracted in 18 minutes. So you basically load the sample onto the sample chamber, and that's it, load it into the machine. It's like, it's inspired by the old cassette tape recorder, and produces the prep automatically. This is the PCR system, and we have these systems out there. If anybody would like to have a look at it. We have a single base system, a 2-base system, a 4-base system, and also a 16-base system that can run samples completely independent of 1 hour of each other. And the actual runtime on the PCR is about 30 minutes. What makes this happen is the chip. And this was something that was inspired by space technology, where low temperature co-fired ceramic is used very extensively. We started with silicon, and then we quickly realized that to scale a silicon chip out of India is going to be a very massive exercise. We still don't have silicon foundries, and today the country is seriously looking at setting up silicon foundries. But we found low temperature co-fired ceramic to be an interesting option. This is a glassy material, it's ceramic, and it allows us to build it layer by layer. It allows us to get very uniform temperature in the well, and therefore prevent any PCR artifacts. That entire chip is the PCR. So you can heat it, you can cool it very, very rapidly. It's in fact so rapid that if required, we can actually run the PCR in 10 minutes, 40 cycles. But we don't do that because all the biology also has to catch up. And of course, you can use fast enzymes, etc. But we are not in a race. And half an hour we thought was a reasonable time to run the PCR. It also has a smart memory built in. So you can't use the wrong chip. You can only use the chip once and all the other things that are associated. The entire lot traceability is built into the chip. The standard curves are built into the chip. And all of this allows for complete traceability in manufacturing. So this is our workflow for stool. You take the stool sample and transfer the swab into our buffer. And we've consistently, because we are a point of care test, been worried about how do we ensure that the sample is biosafe. And we've ensured that with whatever sample that we start with, once it goes into the buffer, it's completely biosafe. So with stool, we gently have to vortex the sample and transfer the clear contents into the cartridge. So that is an additional step, but that can also be achieved by very simple shaking of the tube. You don't necessarily have to vortex the tube. Then you run this on the PCR and you get your results. The power of the platform is actually leveraging mobile connectivity. And all the MolBio systems, all the TrueNAT systems are connected systems. So you can push data real time. They're very useful in a situation where you're monitoring what is happening in the field. You can get real-time analytics with something like this. You can also push updates and other things that are required into the device. You can use it to monitor stock levels, if that is a necessity, and so on. All we require to actually run the PCR is a stool. And this is actually a picture of a lab in Andhra Pradesh that actually runs this. So this is all we need. We don't need anything other than this. We have a solar powered backup possible that can use solar power to run these equipment, to charge these equipment. As I told you, they're battery operated and they run the whole day on a single charge. They can also charge very quickly. In about 3 to 4 hours, they can be fully charged and ready. But if you don't have power also today with solar, we can run it. Specifically with relation to cholera, and this is a semi-quantitative test for detection of the cholera bacteria. And we are using the cholera enterotoxin gene. We have optimized this with a strain from Zeptometrix. We've finished our analytical studies. We have done early studies here at NICED, and those studies are continuing. And that, in short, is my presentation. This was what we have so far with the study at NICED. As I told you, this is an interim data, and the study is continuing. Happy to take any questions.
Thank you. Questions? There's one in the back.
Thank you, Dr. Chandy, for that presentation. I just want to know if you have looked at multiplexing with the PCR from stool samples at all.
So this is an interesting question, and we just had a chat with Andrew on the near. Yes, this is important for us to look at. We do not have something that is done in this area yet, but both within the cholera detection as well as to look at multiplex detection of dysentery. This is something that we must look at. We can do a limited amount of multiplexing with this platform, and that is definitely possible to do.
Another question here.
There was a problem with the RDTs because you had to specially procure them, and this is not being used effectively. In other words, simple but not being used. So you have one advantage, that because of your equipment being used for many other things, it is there in every lab almost, or will be there in every lab. So the business model should be like this, that like what BD do or where Olink and other people do, that bundle reagents which could be procured by people, and they use the equipment by buying the reagents either from you or any of the vendors. So that they will not be dependent on a program or other things. It will easily move. So it is just creating a business model where more cholera tests will be done in the labs wherever there will be a severe diarrhea, severe watery diarrhea.
Yes, sir. You're absolutely right.
Thank you. Oh, sorry, there's one more question. That would be the last.
Thank you for the great presentation. Can you talk a little bit about the cost of the tests?
So
Both the tests and the machines
yes,
themselves.
absolutely. Machines in India are between, I think, 6 and 12 lakhs, depending on what is the configuration that is chosen. National prices may be a little different. The test price purely depends on volume. And if it is a national program based test like TB, that is available at around 640 rupees today to the government. But most tests to the private sector are less than 1,000 rupees.
Sorry, I think we do have to move on. If you don't mind holding your question for the speaker afterwards, you can ask him. But thank you. Thank you, Dr.
Thank you very
Chandrasekhar.
much.
So it's on. Last talk of this session is online, and it will be from Anne-Laure Page, who works on in vitro diagnostics at the WHO, and she'll be talking about WHO PQ systems for in vitro diagnostics.
Good afternoon. I'm Anne-Laure Page, and I'm part of the in vitro diagnostics assessment team in the pre-qualification unit at the. What I would like to present to you today is the general overview of the WHO pre-qualification assessment, focus on the assessment of cholera rapid diagnostic tests, and then open up to what's WHO pre-qualification of in vitro diagnostics is a program that started in 2010. The aim is to promote and facilitate access to safe, appropriate, and affordable diagnostic tests of good quality. And the focus is placed on tests for priority diseases and their suitability for use in resource-limited settings. You can see here on the right-hand side, the current scope, so the current tests that are eligible for pre-qualification assessment, which include HIV, hepatitis B and C, malaria, human papillomavirus, G6PD deficiency, cholera, syphilis. We've just very recently opened up to tuberculosis nucleic acid tests, and we're working on opening the scope to diabetes-related diagnostic tests. So the scope continues to expand, and there are currently 105 individual tests that are pre-qualified, and the list is available on the website that is shown here. The pre-qualification assessment is a comprehensive assessment of individual tests through a standardized procedures aimed at verifying that the product meets WHO pre-qualification requirements. It includes 3 components: the review of a product dossier, performance evaluation, and manufacturing inspection. And the requirements for each of these 3 components need to be met for a product to be pre-qualified. And if these requirements are met, then the last step is a review of the labeling, so the instructions for use and the general kit labeling to ensure that this is. I will give a brief overview of each of these 3 components. And to start with the product dossier, which is a summary of the data assembled by the manufacturer. So the aim of the review of the product dossier is to analyze the relevance of the data provided by the manufacturer. So to make sure that there is quality data that supports the manufacturer's claims of quality, safety, and performance. The dossier is sent to external reviewers, experts in the field, who look at, in particular, the validation studies performed by the manufacturers, and will look at will make sure that these studies are appropriate and well designed. So these include analytical performance studies as well as clinical performance studies. So this is a review of the completeness, accuracy, and consistency of data over the life cycle of the product from the initial design of the test through validation, manufacture, and release onto the market. The experts will verify if the technical specifications are met and if the manufacturer has considered the use of the product in resource limit. The manufacturing site inspection is coordinated by the inspection team in the pre-qualification unit. All sites that are relevant to the IVDs are considered, and the inspectors will generally, through an on-site visit, review the evidence of an implemented quality management system based on international standards. There is also consideration of the robustness of the product for WHO intended settings and use. And the inspectors will also look at the evidence that sufficient capacity is available to ensure reliable production and delivery of the kits if the product is pre-qualified. The performance evaluation is an independent verification of the performance of the IVDs submitted for assessment, and if applicable, it includes a verification that the UN procurement criteria are met if these criteria are available. These are done according to standard methods that are described in the performance evaluation protocols, and there is one protocol per type of IVD. The evaluations are conducted in performance evaluation laboratories, which are laboratories that are audited and listed by WHO to conduct evaluation for one or several types of tests. There are currently 14 listed performance evaluation laboratories, and more specifically for cholera, there are two labs listed, which you will be familiar with because one of them is ICDDRB in Bangladesh and the other one is NICED. For the dossier requirements, so for the pre-qualification of cholera RDTs, the current scope is only cholera rapid diagnostic tests for Vibrio cholerae 01 or 01 and 0139. So PCR tests, for example, are not currently eligible for pre-qualification assessment. The dossier requirements are described in the technical specification series TSS5, which was published in 2018, which is when cholera rapid tests started to be eligible for pre-qualification assessment. So this document describes the data that is required to be provided by the manufacturer, and which include validation studies about acceptable specimen types, about the stability of specimen before testing, repeatability and reproducibility of the test across runs, lots, days, operators, sites, etc. How the manufacturer has validated the reading time of the test, how have they validated the analytical sensitivity, the analytical specificity, which includes the influence of potential endogenous and exogenous interfering substances, as well as cross-reactivity with other relevant pathogens. They include requirements for flex and robustness studies, so to look at the influence of volume, reading time, temperature, and humidity, and light on the result of the test. And there is a part also on the requirements for the clinical performance, which will include a minimum of 100 positive specimens for each serotype claimed, and at least 200 negative specimens from 2 different. Regarding the performance evaluation, so the independent performance evaluation, which is part of this pre-qualification assessment, there is also a protocol available, which was revised in 2022. These evaluations are conducted in either one or both of the listed performance evaluation laboratories. They're using fresh stool specimen that can be left over from routine surveillance or collected purposefully for the evaluation. And the evaluation should include at least 120 positive specimens and 200 negative specimens, which are characterized using culture and PCR. Testing of the specimen is done on 2 different lots of the product under assessment, and it is done. This is the reference algorithm that is used to characterize the specimen. So culture is done initially on selective media with and without enrichment, and specimens that are positive by culture for Vibrio cholerae 01 or 0139 are considered positive without further. While specimens with no presumptive identification of Vibrio cholerae 01 or 0139 are then further characterized by PCR for CTX 01 and 0139, and the final result is considered based on operational characteristics and ease of use are also part of this performance evaluation, and they are reported by the operators who performed the test during the evaluation. These include the assay characteristics, so the number of steps to perform the test, the availability of controls, etc. Some information on specimen collection and storage, safety, kit storage requirements, and equipment and consumables that are needed but not provided. And operators are also asked to provide their opinion on the ease of use of the test, so the clarity of the instructions for use, clarity of the kit packaging and labeling, the ease of use of the test, the ease of reading and interpretation, and an overall. The analysis includes estimation of sensitivity and specificity on each lot compared to the reference algorithm, as well as the assessment of inter-reader variability and inter-lot variability. The analysis and report are prepared by the performance evaluation laboratories and then reviewed by WHO, and a draft report and the data are shared with the manufacturer for review before finalizing the report. The main aim of this evaluation is to independently verify the performance claimed by the manufacturer, and the results are interpreted in conjunction with the GTFCC guidance documents and discussed with the WHO cholera group. If a product is pre-qualified, then the summary of the performance evaluation results will be included in the public report that will be published on the website for all pre-qualified products. So just to open up a little bit this presentation. So first, just to say that there is currently no cholera rapid test that is pre-qualified. The current pipeline, so the tests that are currently under assessment is available on the website. And you can see that currently there is one test under pre-qualification assessment, which is the Crystal VC01 test from Achai Healthcare Private Limited. And again, I wanted to open up to other diarrheal diseases and enteric pathogens, because I know this session is not only about cholera. So as you may have seen on a previous slide, the IVDs for other diarrheal disease and enteric pathogens are not currently eligible for pre-qualification assessment. And we already have a list of tests that will be added in the near future, and it is unlikely that these tests will be part of these next tests. However, we're working on an alternative pathway for assessing the performance and quality of other types of tests that are not eligible for pre-qualification assessment. So this is the expert review panel for diagnostics, which is a risk-based assessment process that includes the desktop review of the manufacturer's data and quality management system. And the outcome is that a risk category is assigned for each product. And then it's up to the procurers to make their decision procuring or not these type of. With this, I would like to thank you for your attention, and I'd be happy to respond to questions. Thank you.
Great, thank you. And Dr. Page is online, so I think we can take a question if there is one for her. Any questions? I think you had a very clear talk. Oh, maybe Nick has a question.
I'll just ask you for more detail, and Laura, about the thoughts on PCR-based molecular diagnostics, especially when you're thinking towards including other enteric pathogens as well. What's the current? You mentioned that that's where you're going. What are your current feelings on PCR-based molecular tests for pre-qualification?
So can you hear me well? Yes, okay. Thank you, and sorry for not being able to join, but I'm happy that I can address this question remotely. So when we had the initial discussions with the cholera program at WHO to identify the need for a pre-qualification assessment, there was a clear focus on rapid diagnostic tests, because this is where it was seen that there might be a wider use of the test in the field, and the need. There were different tests available, and the need to identify which ones are of adequate performance and quality. To my knowledge, there were not many commercialized PCR tests. And PCR was not at the time seen as a first-line diagnostic test for which pre-qualification was needed. But we are in constant discussion with the cholera group, the GTFCC and the cholera team at WHO, and we will follow up with them according to the evolution of the needs, as well as the evolution of the availability of commercial PCR tests. So I think, again, it is, well, we will have to follow up with them, and it is a possibility if there is a clear need identified and a few tests available, we could either find one way or the other. So either through pre-qualification, as cholera is already in scope, or through the ERPD panel, that we could do maybe a more limited review, but still make sure that the products are adequately validated, and that the manufacturers have an adequate quality management system to ensure that the test is of reliable quality.
Thank you very much. Oh, we have a question.
Yeah, just I have two questions. The first one is, what is the yardstick of the pre-qualified kit in terms of sensitivity and specificity? My second question is, you are planning to have the same yardstick for the antibody-based detection kit, as well as Nick mentioned, the molecular diagnostic is coming up in a big way. So the yardsticks are the same for the antigen antibody-based technique as well as for the molecular technique. Thank you.
So I may have missed the word. Just to clarify if I understood the questions correctly, are you asking what are the thresholds and the criteria for pre-qualification in terms of sensitivity and specificity? Is this correct?
Yes, Annie. Yes, Annie.
Okay. So our aim is mostly, is mostly to make sure that we can independently verify the claim from the manufacturer. So in the instruction for use in the manufacturer's data, the manufacturer will say our test has a sensitivity of, I don't know, 90% and specificity of 95%. I'm giving an example. If we will, in our evaluation, we will determine the sensitivity and the specificity. And of course, we're not expecting necessarily to find exactly the same, but we need to find results that are consistent with what the manufacturer claimed. Because what we think, so that in the end, procurers can trust that they know which sensitivity and specificity, that the sensitivity and specificity of that the manufacturer claims is it can be reproduced independently. And because we realize that these tests are not for individual diagnosis, so we're not as strict as what we are for example, for HIV, where we have very strict criteria that the tests need to be at least 99% sensitive, 98% specific. But for cholera, the rapid diagnostic tests are mostly for surveillance purposes. So we realize there's a little bit more flexibility on the sensitivity and the specificity. However, what is important is that the user know the possible limitations. So they know what is the sensitivity and specificity and can adapt maybe their surveillance algorithm, the number of tests to perform based on a known and valid sensitivity and specificity criteria. We are aware also of the guidance documents that were produced by GTFCC, which provides some targets for these performance and of course of the TPP. So we also discuss with the cholera team at WHO to make sure that what we find in terms of sensitivity and specificity is adequate with the programmatic use for these tests.
Can I just ask, Anne-Laure, what is the timeline currently for the ARC-rate test to go through evaluation?
It's a good question. It's ongoing. We have had a little bit of delays in the past years because of the emergency use listing, and so pre-qualification, the emergency use listing of SARS-CoV-2 tests have been prioritized over pre-qualification assessment, and we're now trying to catch up with our activities on pre-qualification. It will not be for this year. So it's a little bit, it will be probably before the end of next year for sure, and hopefully by the middle of next year.
Fantastic. You realize you're on record, Anne-Laure. We do. So we do have one last question then from Dr. Sack.
I wanted to ask the relation between WHO pre-qualification and the availability of the cholera test through the UNICEF supply catalog. I noticed that the Crystal BC and maybe the BioLine is available through the UNICEF catalog, but I was previously under the impression that it had to be pre-qualified in order to be included in the catalog. Can you clarify the relation between the catalog and pre-qualification?
So indeed, my understanding of the situation is that because there is currently no pre-qualified test, however, there's a need for these tests, then this is why there has been another process, which I'm not exactly clear about, on the decision to include some other tests on the catalog. But this is because, again, currently there is no pre-qualified test, and there will be discussions, I believe, once a product hopefully is pre-qualified in the near future, what is the impact on the UNICEF catalog?
Okay, thank you very much for all your questions. And Laura, thank you very much for your talk and for your willingness to come online. Thank you.
Thank you.
So we have, I think, to close out the session, we have some things to give out.
Thank you, Dr. Kumar and Dr. Thompson. And I request you to kindly felicitate today's speakers. May I ask Dr. Arif Tanmoy to please come to the stage for felicitation? Ma'am, please. May I request Dr. Chandrasekhar Nair to please come to the stage for felicitation? Seems like Dr. Nair is not here. Okay. So I request Dr. N.K. Ganguly to felicitate the chairperson of this session, Dr. Kumar and Dr. Thompson. So please. It was a wonderful session. Now we are ready to begin our third session, that is Meet the Experts. I request Dr. N.K. Ganguly sir again, ICMR India, and Dr. J.D. Clements, IVI South Korea, to kindly grace your seats on stage.
So we need just 5 minutes to arrange the stage. Please cooperate.
Assessment in the other room has finished, but Dawn is there.
We are slightly running late, so. না ওই ঘরে বসে আছে এটা এই দুটো কাটা ইনি প্রথমে ঠিক করেছেন আসবেন আবার চলে এসছে আচ্ছা প্রমোদ গর্গ আসেননি এটা পাঠিয়ে দেওয়া ঠিক আছে এক্সপার্টস এরা চেয়ার করবে এরা চলে আসার পর দেন আমি এক এক করে এক্সপার্টসদের টানবো মুশকিলটা হচ্ছে এটা এটা শুধুমাত্র কি করবো যদি চেঞ্জ হয়ে যায় আমার ঠিক করে ডেবিট সাইড দেবে তারপর আমার দেখে বড় মায়া হলো আমি ম্যামকে বললাম উনি বয়স্ক মানুষ তারপরে ইনকে গাঙ্গুলিকে দাও ইনকে গাঙ্গুলি গেলেন আচ্ছা তারপর এখন কি করবো ম্যাম কমপক্ষে 3 থেকে 4 বার চেঞ্জ হয়েছে পরে তো তাই হবে পিছনে যাচ্ছে বলতে So the experts should Dr. Tamir, yes.
Yes, I'm there.
Oh, sorry, sir, because I don't know you.
No problem. So who are there? Dr. M.K. Ganguly is
Dr.
there.
A.K. Ganguly is there.
Dr. Clements, where did you go? Dr. Clements, Dr. Clements.
Most of them are a ma'am. Only very few people. So
You have to find a way, na? Because time
we have asked our ma'am. Ma'am has told us যে উনি inform করবেন, then we will announce and start.
What you can whatever number is there. আগে ওদের থেকে স্টার্ট করে দাও।
Okay, ma'am.
Then they may be asked later.
একটু টাইম দিন, আমি ম্যামের সাথে কথা বলে then I will start. Okay, ma'am. আমি নামে তারা দিচ্ছেন এই ভদ্রলোক বলছেন শুরু করে দাও শুরু করে আমি বললাম যে স্যার
Dr. Khatri is already there.
বলেছে কিন্তু ওনার বক্তব্য অলরেডি উই আর রানিং তোমরা কত দেখি আরেকজন ভদ্রমহিলা আমি চিনি না।
কেউ নেই
উনি বলছেন যে জাজিং বাই দ্য ফেস আই ক্যান সি ডক্টর এনকে গাঙ্গুলি হিয়ার, আই এম অলসো হিয়ার, ডেভিড স্যার ইজ অলসো হিয়ার, হোয়াই ইউ গাইজ আর গোয়িং লেট?
দীপিকা সুরেশ একটা প্যানেল আমি একটা কথা বলবো।
স্যার একটু টাইম দিন, ফিরদৌসী কাদরি ম্যাডাম তো অলরেডি ওখানে চেয়ার করছেন, ওনার শেষ হলেই আমরা ওনাকে নিয়ে এসে শুরু করে দেবো। নাকি সবাই নাকি এই হলে আসছে, মানে অন্য কোনো হল থাকছে না, জাস্ট এই হল, এসি সব এখানে। আমি বলছি যে ব্যাগগুলো নিয়ে আবার ওদিকে যাচ্ছি, বলে যে চেয়ার তাহলে দিতে হবে, বলে না সবাই দাঁড়িয়ে থাকবে, মানে পিছন দিকটা ওভারক্রাউডেড হবে। আমি নেব না, প্রথম দিনে দায়িত্ব নিয়ে আমি ভুল করেছি। শুরু করবো, কি শুরু করবো? এটা আমার আমি মানে
অ্যানাউন্স করতে কলেজের শেষ হলে এখানে স্টার্ট হবে।
এইটাই তো মুশকিল হয়ে দাঁড়িয়েছে না, বিকজ কেউ জানে না হলেতে কখন শেষ হবে। ম্যাম তো বলছি, ম্যাম সুস্মিতা দুজনেই বলছেন উনি এত কেন বকবক করছেন, শেষ না হলে কোথাও আমি লোককে নিয়ে আসবো, কেউ ওনারা গেছেন। শুরু করবো, আমি কথা বলা শুরু করছি। Explain or give you an insight because I myself don't understand. কিছু লোক তো চিনি, ফিরদৌসী কাদরিকে চিনি, গগনদীপ কান্ত, ফিরদৌসী কাদরি, গগনদীপ কান্ত চিনি।
এটা কি আসছেন?
এইটা এটা কি রায়ান? ইনি হাত দিয়ে দেখানো, সরি। ইনি কে? এনার নাম কি আছে? চিনি না। দীপিকা সুরকে মাইসের থেকে এসছেন, কে চেনো তোমরা? ওনারা এখানে expert। ওনারা যদি বলে যে আমার সাথে 3-4 জন এসে করতে শুরু করে। না আমি ছিলাম না বাথরুমে ছিলিস তো যখন আমি কথা বলছি না আমি যখন তুমি যখন স্যারের সঙ্গে ছিলে তখন আমি বোধহয় খুলতে গেলাম ঠিক আছে ক্লিক করেছি কিনা জানি না তবে এত সামনে লোকজন ছিল তোমাকে দেখা যাচ্ছে না তুমি ছিলে না আমি তাক করেছিলাম এই লোকজন এমন সামনে এবার তো করিস চেষ্টা করিস উঠবো না আমি এখানে দাঁড়িয়ে নাকি তুমি আর তোরা কি কেউ ফটো তুলতে পেরেছিস আমার আচ্ছা তখন সঙ্গীত তুলতে পারে ছিল না আমি এখানে দাঁড়াচ্ছি হ্যাঁ হ্যাঁ স্যার মোটামুটি সবাই কি এসে গেছেন আমি এখান থেকে শুরু করছি সবাই কি এসে গেছেন আমি কি শুরু করবো নিচ থেকে ডাকছি হ্যালো Hello. We are ready to begin our third session, that is Meet the Experts. May I request Dr. N.K. Ganguly, ICMR India, and Dr. J.D. Clements, IVI South Korea, to kindly grace your seats on stage. I also take the liberty to call the expert of this session to kindly come up on stage and take their seats. The respective experts are Dr. David Sack, John Hopkins School of Public Health, USA, Dr. Firdausi Kadri, ICDDRB, Bangladesh, Dr. Edward T. Ryan, Harvard Medical School, USA, Dr. Gagandeep Kang, Christian Medical College, Vellore, India, Dr. Tamid Ahmed, ICDDRB, Bangladesh, Dr. S.K. Bhattacharya, ICMR NICED, India, Dr. Uday Ghoshal, SGPG IMS, India, Dr. Deepika Shur, ICMR NICED, India. I request Dr. Ganguly and Dr. Clements, sir, to lead this session.
We are waiting for John, but here there are some major expertise riding over here. On my right, on my right is Dr. Firdausi Kadri. In ICDDRB, she is fondly known as APA. So she is an immunologist who has contributed immensely in the vaccine trials, vaccine development, vaccine evaluation. Almost every vaccine which were evaluated in ICDDRB has her fingerprint. So I didn't know how the session will go, but what is expected that you will all ask any question which is bothering you, and if the expert is able to answer, they will answer it, and if there is no answer, they will say that we do not know. So then we have Dr. Dr. Sack. Dr. Sack is has been the former. There are now 3 ICDRB. The panel is dominated by ICDRB guys. So Dr. Sack was a former director in ICDRB, and we have Dr. Clemens, another director of ICDRB, and Dr. Tahmid Ahmed, current director of ICDRB. So we have Dr. Ryan, we have now joined by Dr. Clemens, and we have Dr. Ghoshal. So, and we have Dr. Deepika Soor, who did the cholera trial in the DOMI project, which Dr. Clemens brought to Calcutta. She was the front runner over there. So with these, I will make some opening comment and then give John the mic to make some opening comment. And then there are 2 methods. We can ask each one of them of what they feel is important, and then you could shoot the questions. So let me start with myself. Today I remember Dr. Jyotirmay Das, who attempted the first time to do the physical mapping of the cholera vibrio. He did, and he succeeded also, but he didn't know that cholera has got 2 chromosomes. Vibrio, Dr. Honda has done that. And so that particular effort was later on done by others, but it was the first in India. And then the same group in IICB, Dr. Amit Ghosh, created the moving element of the genome of the cholera vibrio. And Dr. Ranjit Ghosh, who is now in USA, also worked on the same area in IICB. So these guys brought an immense value in the genomics and genetics of cholera. And today we heard how the Yemeni strains have changed and how the drug resistance element work and influence the system. My second comment, which will be very important for enteric pathogen, because I have started working on that and I find it very rewarding. There's the multi-omic approach for diagnosis, for prediction, for outcome, as well as designing the products, whether vaccine or a protocol. And here the few very new platforms have come up, apart from the metabolomics, immunomics, proteomics, and the genomics. There is a lipidomics. There is a link now with the O-link, the new targeted proteomics. There is now a link between genomics and proteomics. And that provides a lot of answers which were not available before. There are 4 or 5 very major papers on the infections. Like Henning Wedemeyer has worked out very nicely the hepatitis C profile. The same way Dr. Ujjwal Neogi of Karolinska has done for HIV. And the Pankaj Seth of an NRBC has done for the COVID. So there is a huge scope of applying this science in the area of enteric region. So this is an area where I find very few people are working for enteric pathogens. It will really be rewarded. Now, with this opening comment, I give to John to make few opening comments, and then we will pass on to everyone to make a 1 minute or 2 minutes opening comment.
Well, after Professor Ganguly's comments, I often think back to a board meeting we had at ICDRB in which Dr. Ganguly was making one of his incredibly erudite comments. And Professor Ian Gust was asked to say something. Ian, a very eminent Australian virologist. And Ian said that after Professor Ganguly talks, I always feel like the ninth husband of Zsa Zsa Gabor. So I think I guess my comment is not so much scientific. It's more ASCOD and more about what I'm hoping that ASCOD will do, and that is helping to build the next generation of scientists in this field, and to create a venue for the establishment of relationships, because I think all of us in this panel have discovered that our careers have been extraordinarily dependent on and benefited by our relationships with other scientists. Science is now a team sport. And I think back to my very first ASCOD meeting, which I think, David, when was it, 84? That I don't know, I'm not sure it was 1884 or not. And in those days, I was a young scientist at CDRB. David was my boss. We were doing the cholera vaccine trial together. At any rate, we went to one of the first, not the maybe the second or third ASCOD meeting in Bangkok, and I remember at one of the coffee breaks meeting an Indian, a young Indian scientist who was about my age, and we had not known each other before. We struck up a conversation. He asked me what I was doing. I mentioned the cholera vaccine trial. I asked him what he was doing, and he said that, well, he was working with Dr. John Robbins at NIH on his then VI polysaccharide vaccine. Well, it turns out that scientist with whom I then developed a lifelong friendship and collaboration was Professor M.K. Bond. And I'm very hopeful that you all in the audience will use this really unique opportunity to develop personal and scientific relationships, and they're often intertwined, to advance your careers in this field. Thank you.
Thank you, John. Actually, John thrives on the scientific relationships. So we will first finish with the former or current ICDDRB directors, executive directors. So, John, did you have any disagreements with John Clements?
Of course. I mean, what would scientists be if we didn't have disagreements? That's how we settle. We have different opinions, and then we get data. So it's important that we, that we have discussions, we have disagreements, and then if we have disagreements, we go out and get data and settle them. I would like to just follow up with what John said. After working in Bangladesh for, I guess, a total of about 15 years, during the last decade or decade and a half, I've been working in Africa, different countries in Africa. And I think about the benefit of ASCOD in Asia and the lack of a similar kind of conference in Africa. We have good scientists in Africa. I know many of them, but we don't have the group that forms ASCOD, and we don't have the relationships between countries that you have here in Asia. Perhaps that's a next item on my agenda is to see, is there a way to form this kind of a conference format for the scientists of Africa?
Agree with you, David, that this was in the ASCOT agenda, and Dr. Kadri will perhaps move it. And we have some karaoke, we have some important people who have contributed immensely in the area of. Now I go to Tamid Ahmad. He has put the nutrition into a completely different plane. When I was talking that in the multiomics are not being very popular with the scientists in this area, I eat my words because he published a paper in Science using multiomic platforms for the nutrition. So I give him the mic now.
Thank you, Professor Ganguly. What a great honor. I have three bosses here, as my co-panelists, Dr. Sack, Dr. Clemens, and Dr. Ganguly himself. So it's both an honor and in a way, little bit of intimidation, okay? But, again, I must say that this is a great, great occasion to talk about cholera, to talk about enteric infections, including other types of diarrhea, clinical diarrhea. I must tell you one thing, little bit before this meeting, I was having a chat with Dr. Shumon Kanungo, and he was asking me, what do I think about cholera beyond, okay? Immediately, my thoughts went back to Bangladesh. Bangladesh and West Bengal, these are hubs of cholera. But do you know that when I talk with my friends, with my family, sometimes they ask, do we still have cholera? So that's the perception. And the perception is because talking about cholera is almost like a taboo in a country like Bangladesh. But the months of May, April, May, changed everything. We had the largest cholera outbreak, epidemic in the country. In a hospital that can take care of only 300, 350 patients per day, the hospital run by ICDDRB had to take care of 1,500 patients per day. Imagine. A total of about 60,000 patients coming to the hospital, 40% of them coming with severe dehydration, in shock, just seconds away from death. And this happened in an age when we have this James Webb telescope sending images billions of light years away. Imagine what is science doing today, and still we have cholera ravaging countries like India and Bangladesh. So this thing has taught us many things. One is that we really need to look at the social determinants of cholera. This is something that we do at ASCOT, but I think we should do more of it. Because cholera to me is a disease of development. If you are developed, you don't have cholera. So that brings to our mind the social determinants. We really need to look at that. A week ago, the New York Times, called me and they had an interview. They asked me, why don't we have this cholera vaccines now? Why is this shortfall? Why is the WHO talking about 1 dose and not 2 doses? And my response was that look, we need to have these vaccines all over the world where we have cholera. We need to have these factories, pharmaceutical companies in India, in Bangladesh, in other parts of the world. We need to have this pre-qualification stuff done, expeditiously. Until unless we can't do it, we won't have the vaccines where they are required. So today, I would say that we need to talk about cholera in a very social way, because this has huge social ramifications. That's why we still have cholera, and we need to do something for the coverage of this oral cholera vaccine wherever it is needed. Sir, I'll stop here and thank you.
Bring in NICED. So Dr. Bhattacharya was the former director of NICED, and he was the first regular director of Dr. Paul, and is responsible for the infrastructure which the NICED now boasts of. And he is a clinician. So, but clinician research scientist, and has got all the laurels. He's a fellow of National Science Academy. So he did science, but it is a rare mixture of being a clinician and developing the clinical science in an organization. And whatever he touched, it almost turned gold. So I will give Dr. Bhattacharya the mic to make.
Thank you, Professor Ganguly, for your nice words. I'm very pleased that the scientists with whom I worked during the last 40 years, many of them are here. I know Dr. David Sack, Mrs. Kadri, John, Deepika, and Sir is our mentor, Professor Ganguly. So I'm here. One thing I want to emphasize here, that when WHO found out the WHO recommended formula for WADES, there was a need. They established that it is effective, was established. But what was needed is its dissemination and implementation at the community level. That was done by Dr. Dilip Mahalanobis. Unfortunately, he died 1 week back. But one scientist whose name nobody talks about that is Professor S.C. Pal, the then director of NICED. He got a grant from WHO, and with that grant, he formed a team, a clinician, a gastroenterologist, a microbiologist, and a pediatrician, and he himself. And these lectures consisted of epidemiology, management, prevention, and other aspects. So we used to travel across the country and also abroad and train the pediatricians and internists about the implementation of oral dehydration solution for dehydrated patients. Then came how to prevent dehydration when diarrhea ensues. This remarkable work of Dr. Pal has never been recognized. And I will be very happy, and I appeal to this gathering that let's have a small dialogue about the contribution of Dr. Pal in this conference. And he and Professor Ganguly attended, worked out the first Asian conference on diarrheal diseases in Dhaka. The other aspect I will emphasize is that any medical doctor who is doing research, biomedical research, must have some exposure in the clinics. They must have a responsibility of working in the hospital or in the outpatient department, as well as conduct research. If these two interfaces cannot be linked together, I believe the researcher is not complete. This is my experience over the last 20 years when I left research. At this juncture, I believe this intermingling is necessary. Thank you. Thank you very much.
Our school is a very important place.
But we bow to Hopkins. Yes, so.
But he has got science and thinking on how the future cholera vaccines should look. And it was very fascinating to hear, and the data which they have generated. But it is not only the data generation, how to create assays which will actually prove that was something very, very nice and futuristic. So I give Dr. Rand the mic.
Well, it is a wonderful opportunity to be with everybody. I tend to be an intrinsic optimist. And so I often, ASCOT is one of my favorite meetings. And I often use meetings as milestones, not just to see people, but also to take stock of where we are, what we've accomplished, what needs to be done. And I was not at the original ASCOT in 1981. But if I think of over those 16 ASCOTs that have occurred over the last 40 odd years, and I think we should recognize all the accomplishments, not only by the people here, but all the people who've gone before us. And if I think on the cholera side, the task force has been reinvigorated, the global stockpile, Gavi has been formed, OCVs have rolled out, WASH has made significant advances around the globe. And yes, we still have a significant cholera problem. And yes, we have a lot of work to do. But also in that time period, probably over 2 billion people have joined our species on this planet. And we're fighting it, not fighting against that, but that's the reality you have to deal with. On the typhoid side, the typhoid conjugate vaccine has rolled out. The epidemiological modeling has really hit its stride and doing sort of computer analysis for identifying risk factors and interventions, et cetera. On the nutrition side, individuals like Dr. Ahmed have made significant contributions in advancing the health of young children. So yes, we have a lot of work to do, but also it's an honor to work with so many people. In my family, we have a philosophy that God has no hands on this planet but ours, that God has no feet on this planet but ours. So if we want to get something done, we need to do it. And it's wonderful working with all your hands and all your feet over the last years and hopefully going forward in the future. No.
Oh, Dr. Kadri, the president of ASCOT.
Thank you. I'm glad you didn't give it to me saying ladies first. That's something that I don't like. So where do I start? I think I've learned so much in life from research and from collaboration. So I would like to talk about a few things like cholera, typhoid. 40 years we've been saying, let us have a vaccine, let us do this, let us do that. We do have vaccines now for both for cholera and typhoid. Now, the time has come for us to know more about these diseases even more. When I joined ICT-DRB, and I wanted to work on cholera, Tahmid, if you remember, people said, David was there, my division director. They said, don't think you said it, but the board said it, that cholera ORS is there, a vaccine has been made, no research needed until 0139 broke out. And then I came out of Shigella very quickly, very happily, and started working on cholera. And to this day, I think it has been a wonderful area to work in, starting from the omics to the immunology, to everything and to the downstream process. It's such a challenge to get up every day and find things to do, and also typhoid. I mean, if we don't do things for our own settings, who will do it? We can get all the help we want. I have the greatest friends over here on this dais, left, right, everywhere. In Asia, in the USA, in Sweden, everywhere. You just have to stretch your hand out and say, I want to work on this, and you get the help. So we have all these young people over here, and also mid-level. And we sometimes feel frustrated about, how do we go? Where do we go? But I think the best thing is to have a passion for something. Like my passion is to find solutions. And at ICT-DRB, you cannot forget that, because every 5 months you start planning for the board or the SAG, and you're asked, what have you produced? So that keeps us on our toes. But everywhere you don't have the same thing. But I would say that we must have knowledge at every step. The high throughput is very important. The basic is very important. But the applied is very important for us in developing countries. How do we implement it? How do we do something to make life better for people around us? But in that way, we also contribute, we interact. And I think if you don't hold hands, if you don't meet people, if you don't come to meetings like this and make use of it, I have had the greatest opportunities in my life because I could see Jim somewhere. Is Jim Fleckenstein somewhere? I remember Ed, when I can just give some. Ed and I were both at ICT-DRB, and Ed didn't know about me and I didn't know about him. He was working at the Clinical Sciences Division, and David Sack told, or Brad Sack told him one day that he was trying to do something in immunology. Have you met Dr. Kadri? Who is she? And then the day before he was leaving, or almost like that, he came up to my lab and just said, and then that's how we started collaborating. I was weighing out something on a balance and he came and said, hi, Dr. Kadri, you know, and then we started talking, and that's how we've been working for how many years, Ed? 30, yeah, or maybe more. And we've had, we want to be careful about not being too old, yeah, especially me. And so we've been doing so many things, but best of all, I'm also John Yu over here. The worst part is if you, David Yu over here, and our great Professor Ganguly, always helpful. So what I would say is that the only way we can work in science in our worlds is to collaborate, is to use high throughput and low throughput, both. Try to use things so that it can be implemented, and always have a questioning mind and never give up. I think, and always have friends. If you find it very difficult to work with someone, try to find a way to work with that person, but keep on working. I think, what do you say, John? And you become friends. So I think that's my message. I mean, the world is a beautiful place. There's so much to do. We know about climate change. We know about salinity. We know crops are not growing, but we'll find solutions. That's my way of looking at things. Thank you, Professor Ganguly.
Thank you, Dr. Kadri. And I will share a secret about her. She is very helpful to people, but she not only got some huge awards, she also got some huge money out of those awards. And but she didn't use it personally. She had opened an NGO, and that NGO does very nice work. And I remember her as a lady who feeds me Hilsa fish.
Thank you. It is really nice to come to ASCOD, because ASCOD is not only a conference of diarrheal disease, but it brought me back to home for a few days, the sweet home. But let me tell you, we have been talking about relationship. I remember my relationship with Professor Ganguly. He was my teacher 35 years ago. So I wanted to be a clinician in my life, seeing only patients. But we got exposed to some teacher who make metamorphosis in the student. So I can name Professor Ganguly, I can name Professor R.C. Mahajan, I can name Professor S.R. Nayak. So after getting exposed to them, after being taught from them about 35 years ago, I changed my mind and I wanted to be a clinician scientist and not merely clinician. And that is very, very important. Now we are talking about cholera. Now everybody spoke about prevention of cholera. It is very, very important. You know why? Cholera takes away lives. You go to the rural areas, we are talking about oral rehydration solution. We are talking about IV fluid. But let me tell you, even 10 years ago, I have seen death of a young girl due to cholera in my village. So therefore, implementation, practical implementation of science is very, very important. Also, I'll speak something beyond cholera. That means somebody has an acute diarrheal illness. He gets admitted, like Dr. Tamid was saying, because of diarrheal diseases. Now he gets oral rehydration solution or IV fluid, and now he recovers and he forgets about the illness. But it is not so. You see, if somebody gets acute infective gastroenteritis, 10% can have long-term sequelae, which most of the scientists don't bother about. And this long-term sequelae could be either post-infection irritable bowel syndrome or post-infection malabsorption syndrome. Now we have all the directors sitting here of ICDDRB. And obviously I should talk my relationship with ICDDRB. I work in Bangladesh in collaboration with scientists, including Dr. Tamid. And we wanted to see those who develop acute gastroenteritis during long-term follow-up, do they develop chronic sequelae? And to our utter surprise, we found 10% patient who had acute gastroenteritis can continue to have long-term GI symptom, long-term problem in their tummy, and this may be either malabsorption or maybe chronic GI illness called irritable bowel syndrome. What is the practical consequence of this? Practical consequences, we know the scarcity of the food in the developing world. The poor people don't have enough to eat. But we blame the obesity of the West and the leanness or malnutrition of the East entirely related to food availability. But we are realizing it is not entirely so. Because if we are eating, part of the food gets malabsorbed, and the malabsorption can increase if somebody got an acute infective gastroenteritis, which led to protracted inflammation of the gut because of the lack of T regulatory system. And therefore, there is a chronic inflammation in the gut. And that lead to malabsorption of the limited food that the fellow has. And this may be one of the cause of malnutrition, and the malnutrition is not entirely related to the food availability. Now, what happens is we have been working with gut microbiota using culture-based technique for last 2 decades. And I tell you, I'm lucky because my wife is a microbiologist to whom Dr. Ganguly was talking about. So we used to culture the gut bacteria using a special technology by development of a double lumen catheter. But today, whole world is talking about gut microbiota with a different technology, that is next generation sequencing. And we are realizing more and more that the microbiota dysbiosis can be responsible for obesity or malnutrition. And therefore, this is one area which we should be talking about. All the youngsters here, you should talk about prevention of cholera. You should talk about implementation of the cholera vaccination so that we don't develop cholera. But don't think it is going to only prevent the mortality due to acute illness, but also it is going to improve the quality of life of people, preventing malnutrition in the long run, because cholera can have long-term consequences, and that is called post-infection irritable bowel syndrome and post-infection malabsorption syndrome. But also you have to realize some of the scientific thing may not be entirely true, as we realize with time, because nowadays we are talking about a lot of case control studies where you just look at gut microbiota by next generation sequencing of the fecal material among the healthy control and disease population, and you say this is causing this. It may not be entirely true, because disease population has so many confounders which can alter the gut microbiota, including the drugs they are taking, the diet they are changing, the activity level that is happening. So all this can also change the fecal microbiota. So these are some of the things are epiphenomenon rather than really being true. So what happens is it is a great opportunity to talk with you. I love science. I'm passionate about science. I want to bring application in the science so that I can benefit my patient and also understand whatever data come in the world, which data I should pick up for my patient care and which data I should wait till more data come, and then I see whether that was a junk science or real science. Thank you so much.
Bringing out microbiota here, there's a very large number of scientists who are attending this. Bhavtosh is one of them. And I know by my own experience, I collaborated with him to find out responders and non-responders of immunotherapy with metabolome signatures, and it came out so well that those who have a kind of bugs, they responded, and with another kind of bug, the group didn't respond. So now my last person in the group, Dr. Deepika Soor, not the last person really, and she actually did 2 very major trials here. She is a public health person, but the contribution she has done that set up a very good trial, clinical trial groups in NICED. It was not existing after Dr. Pal went. So Dr. Clemens helped to establish that. They said that both the VI trial as well as the oral cholera vaccine trial, both were very successfully done and done in the world standard. So Deepika got a name on this and she pursued this particular thing. But Deepika is also known as a girl who never listens to me, whatever I say. So after Deepika, the mic goes to Dr. Clemens for conducting the rest of this question answer session from audience.
So I really don't know how to answer your comment, but actually I would really like to sort of tell you all so many incidences that happened during our studies, during all these clinical trials, because that would give you all an idea of how difficult it is and also how important it is to conduct these studies in such a perfect manner. But anyway, we don't have that much time. And after all the advice that you all have got, digest it. And all I will say is that you all realize that so much work has been done on cholera and typhoid, but so much more is left to be done. And that now it depends on the younger generation as you all to continue with the work and to develop it further. That's all I'll have to say.
So how much time do we have left?
Oh, yeah, it's already late. It starts at 7.
Oh, we are. Okay. I was going to open this up to questions, but it sounds as if
Go ahead, go ahead.
my sister here is telling me no.
No, but then there might be.
Does anybody have any comments or questions? This is a unique, oh, yes, okay. Is there a roving microphone?
The young one, there are so many microphones.
I can narrate a personal example. I just happened to meet one of the presidents of India, very knowledgeable person, APJ Abdul Kalam. And when he came to know that I work in cholera, he said, "You're wasting your time, cholera doesn't exist." And remember, he was a president of India and a very widely respected person for his knowledge. This happened when I was in Gandhinagar. He came there to inaugurate our institute. And when I mentioned this, that was his comment. Even Professor P.M. Bhargav told me the same thing, but being a very different person in GRC, I took him aside and explained to him. And he said, "Amit, this is information to me I never knew." Imagine P.M. Bhargav, who was a moving encyclopedia. So unless there is a realization on the in the powers that be, it's a real problem, as he said, that it will be very difficult to eradicate this.
I would like to add here that clinicians should also be involved. Very few clinicians, Dr. Nair and I, we had gone all over India talking about the cholera and the vaccine, but the clinicians just denied that they see cholera cases. But then when you ask them that, have you ever cultured the stool? No, we just give antibiotics and they get cured. That's all they have to say. So see how many cases we are missing. So clinicians should also be sort of educated on cholera.
Other questions, comments? Okay. Oh, yes, we do. Okay.
I thought to have the great opportunity to ask a very practical question as a representative, I think, from the young generation that I usually heard. And when I talk with my fellow mates, so there is a dilemma among the youngs when they aspire to be someone like Dr. Kadri or Dr. Tahmid or John. But how to balance between getting an experience and an academic career, right? So when someone is too much into doing field research and getting great experiences, then after 10 or 12 years, they think like, okay, I have enough experience, but I do not have a PhD. So there are fellows who are doing PhD 10 years before than me, and I'm just getting experiences. So as an expert and as a star that you have already established in their own field and created example. So when people like us are confused and with this duality, what to do, in which path should we follow? What is your advice for us?
So it's difficult, but I guess the first is follow your heart. What gets you out of bed in the morning to figure out what to do? And then remember that degrees are just degrees. So you have to figure out what skill set do I want to get and how can I bring that to bear? So the first is you probably shouldn't be spending 12 years getting field experience before deciding to go for a PhD. Probably after 2 or 3 years, if you say, this really gets me out of bed in the morning, and I think getting a PhD will hone my skill set, that might be a time period to move into that. And then you're right. I mean, and this is true no matter what you go into, it's hard to balance, it's hard to keep support going. And my advice for you on that is pick something that you love, work hard at it, make wonderful friends and collaborators, because as was mentioned, science is not an individual thing anymore. And also, if you associate with centers of excellence, usually there's enough of a pathway there that you can move forward with that as well. That doesn't mean don't go off and do something by yourself. That might be the best thing for you. But I think if you do those things, centers of excellence, something you're passionate about, and you work with great colleagues, then usually things will move forward in the right direction.
Yeah, and I think here's also, I think I can comment, like, do not, just follow a goal looking forward like what others are doing. I think goals should be with your own perspective, not like what your friends are doing, what your other colleagues are doing, right?
Yes, that is correct.
Thank you.
I have just small comment. You see, while attending this conference, what you are getting, the knowledge from the presentations, and simultaneously you are making friends with your senior colleagues and others. So these senior colleagues are a kind of mentors for you. And if you can develop a good rapport with these mentors, attending different conferences, get a word from them, refer to certain places. You go there, I will speak to the person, you have a better chance.
I wanted to come back to something that Dr. Bhattacharya mentioned regarding Dr. Paul, and tie that in with what Dr. Ryan is doing. For those of you who don't remember Dr. Paul, at that time, the oral cholera vaccine was in the process of being developed and tested. Dr. Paul was very enthusiastic about the injected vaccine, and he was sure that a better injected vaccine could be very successful. But unfortunately, he was not able to pursue that. And I have a feeling that we may be coming full circle. Yeah, and I think this sometimes happens, that we have to relearn things that we knew years ago, but re-examine and see maybe there's some knowledge from the past that can be applied today.
Any other comments or questions? Yes.
My one question to Dr. Ghosa. So when you are talking that the post-cholera complication, so have you any experience that if the complicated patients been treated with a combination of probiotic and prebiotic, does that improve their health?
Yeah, so you asked a very important question. I tell you, this is still in the infancy. Like during acute diarrhea, you get microbiota dysbiosis, but the severity of the disease is not at all related to that. Also, the long-term consequences are not entirely related to severity of acute gastroenteritis. What you are suggesting is that if we give probiotic or antibiotic, will the long-term consequences decrease? In fact, data is converse or reverse. People have shown that those who had acute gastroenteritis and were given antibiotic more often develop long-term consequences than those who were not given antibiotic. But there I suspect it is possible that there was a confounder. Those who had severe acute gastroenteritis were probably given antibiotic and not those who did not have. But your suggestion, would probiotic prevent long-term consequences? I tend to believe it may be, but currently there is no data at all.
Thank you, sir. So my second question to Professor John. So you know that particularly the endemic regions, the kids those are living in the slum, their gut have also toxigenic tuberculosis, but they don't develop the disease. Why? If the bug is there, if they can produce the toxin, what's prevent them to monitor the disease?
He's asking Dr. John. He's asking.
Yes, I'm asking you, sir.
Can you repeat?
So what I'm asking, particularly the kids, those are living in the slum area, in cholera endemic area. So when we did our microbiome study, we observed that almost 30% of the kids, they have toxigenic tuberculosis in their gut, but they do not develop the disease. They never develop the disease cholera. They're always asymptomatic. What could be the possible reason for that?
Yeah, I think your question is again very important. I will actually address this. See, when we talk about occurrence of a disease, it is not entirely related to the agent. It is partly related to host, and it is in a large way related to host-agent interaction. So you saw corona epidemic recently. One fellow get COVID-19 and he remains asymptomatic. Another fellow gets severe disease. And this is largely related to how the host responded to the agent and not entirely related to the agent or its genotype or anything. So what happens is, yes, to any infection, in response to any infection, there is two types of host response. One type is severe, too much immune attack, killing the own body of the host. And another type is that you just do a controlled sort of immune activation so that you clear the agent, but do not kill the host. So what happens is this you are talking about, that those who had cholera, pathogenic cholera inside their gut, but they did not have the disease, they have a very good T regulatory system. They have a different microbiota. Their host genotype may be different, and altogether they are tolerating the agent. But let me tell you, though clinically it looks like they are tolerating, people have shown many of them develop subclinical or clinical malabsorption. They lose calorie and they become emaciated. And even the doctor do not incriminate that what is happening, that emaciation is related to pathogenic sort of organism in the gut. But we are realizing that more and more. Dr. Tamid will agree with me.
Thank you, sir.
Agree with you, and giving the mic to Dr. Tamid. He has a tremendous work on this area with Jeffrey Gordon, and it is practically now being used also in his preparation.
Thank you, Professor Ganguly. In fact, I'm going to talk about, gut microbiota, childhood malnutrition, and what do we do about it. That's day after tomorrow in the morning session. But just wanted to touch back on what we heard from Asma. And I was thinking about when Ed said that, follow your brains. I think it should be follow your brain and then follow your heart, use your brains and utilize your time properly. And, just to tell you what happened to me, I was all along a clinician. As a clinician, I realized when I was young, if I don't learn certain things, I probably will never be able to learn. One such thing is the ECG, the electrocardiogram. Similarly, in research, as a young researcher, if you don't learn the basic things, the basics of research methods, perhaps it may become too late as you grow. So that would be some of the messages that I can give you, That's it. Thank you.
Said that you give the
So we, during the last very stimulating 5 minutes, we've had about 5 messages that the dance ceremony is eagerly awaiting the audience. And so let me thank the participants on the dais, the wonderful audience participants, a very nice session, a very nice blend of scientific and non-scientific comments. And I wish you great enjoyment for the forthcoming dance ceremony.
Hello. It was a very interactive and informational session. I thank all the experts. Now I request Dr. Ramamurthy sir to kindly felicitate all the dignitaries. And I am requesting volunteers to assist Ramamurthy sir. Dr. Ganguly sir. Dr. Ganguly sir. Dr. Clemens. Thank you all. We end our second session of day 1 here. Please enjoy the cultural program and dinner afterwards. Thank you. We will again meet tomorrow here.